Uh, Good morning, ma'am. We are starting from here.
हाँ
Uh,
होगा ना।
Good morning,
और दूसरी
everyone.
चीज ये है, जब पंजाब में जब पेपर फार्मेसी का लीक हुआ है उस होगा।
Good morning, everyone. A very warm welcome to all our respected dignitaries, esteemed speakers, faculty members,
Good
research
morning.
scholars. Good morning, ma'am. And the participants today for of our faculty development program. We hope you had an enriching learning experience over the last 3 days. As we move into another exciting day, we continue our journey of exploring cutting edge technologies that are transforming pharmaceutical education, healthcare, and research. This session will provide valuable insight into AI is accelerating precision medicine, supporting clinical decision making, streamlining drug discovery, and driving innovation across the pharmaceutical and healthcare sectors. I am confident that today's deliberations will further broaden your knowledge and inspire new perspective in teaching, research, and professional practice. It is now my immense pleasure to introduce our speaker for the first technical session of day 4, Professor Dr. Zeenat Iqbal, Department of Pharmaceutics, School of Pharmaceutical Education and Research, Jamia Hamdard, New Delhi. Ma'am is an internationally acclaimed academician, researcher, and mentor who has made remarkable contribution to the field of pharmaceutics and novel drug delivery system. She is the recipient of the prestigious Best Researcher Award 2025 in recognition of her exemplary and outstanding contribution to pharmaceutical research by P.R.S. Educational Trust. With an impressive academic portfolio of more than 293 peer-reviewed research publications, 47 book chapters, and 8 patents, Ma'am has significantly advanced pharmaceutical sciences through her pioneer research and innovation. She has been consistently recognized among the world's top 2% scientists for 4 consecutive years from 2022 to 2025 in the globally renowned list released by Stanford University, USA, in collaboration with Elsevier, reflecting the global impact of her research. Her academic excellence has also been acknowledged through 42 prestigious awards and honors. With a Google Scholar H index of 49 and an I-10 index of 134, her research continues to influence scientists and researchers across the world. As a dedicated mentor, she has successfully guided numerous MPharm and PhD scholars, nurturing the next generation of pharmaceutical research. Beyond her outstanding research achievement, Ma'am has played a transformative role in academic leadership by initiating several landmark activities at Jamia Hamdard, including Pharmacon, the Spima K Jamia Hamdard Heritage Club, and the annual Nobel Symposium. Her unwavering commitment to academic excellence, research, innovation, and holistic student development continues to inspire the entire pharmaceutical fraternity. Today's session is on the topic of pharmaceutical intelligence in personalized medicine and clinical decision support. We are truly honored and delighted to have Professor Dr. Zeenat Iqbal ma'am with us today to share her invaluable knowledge, experience, and expertise with us. Thank you so much, ma'am, for joining us.
Thank you so much, dear. It is always a pleasure to be with you and listening to you. And Bismillahirrahmanirrahim. Assalamu alaikum and a very good morning to all of you. I bring with me a lot of blessings for each one of you who have participated. And I am extremely happy to be a part of this very important faculty development program. As she has already told, I am here to only share whatever little I have learned on this topic. This is, I must acknowledge in the beginning that doing a presentation on something related to AI is always a unsurmountable task. It is actually very difficult. But then it is helping me to develop a sense of what is required in this 21st century and what this AI is all about. So thank you so much, Dr. Akhil, and all the organizers to give me this opportunity. I'll be very happy to share the little information and knowledge which I can with my very worthy colleagues. And I'll just take permission for sharing my presentation. Shall I go ahead?
Please, ma'am, share,
Please,
ma'am.
ma'am, share. Okay. I trust I'm audible. आवाज आ रही है सबको ठीक से?
Yes, ma'am.
Yes, ma'am. ठीक है. स्लाइड दिख रही है आपको? एक
Yes,
सेकंड
ma'am.
लगेगा. Just a minute. Just a
हाँ.
minute. Just a minute. Am I able to show the slide? The first title slide is visible to all of you?
Yes, ma'am. Yes, ma'am.
Okay. So as she has already delineated my topic, I'll be talking about artificial intelligence in personalized medicine and clinical decision support. And we are trying to understand how we have moved from the population level guidelines to patient level precision. I think as this will be the flow of my presentation, I'll try to stick to my time and maybe a little ahead of that. We have this MPharm examination ongoing, so my presence is required there as well. I am very open for any discussions at the end, and if at all there is anything which is required to be understood at that point of time, I would not mind if you just ask me to stop and explain anything which is not explained. So this first slide is something which everybody of us will relate to. I think the day the human body was born, the disease was born as well. And disease and a human body system is a lifelong companion. There has always been a dynamism which has existed between the environment, between the biological and genetic factors, the lifestyle and environmental factors, and psychosocial and behavioral factors. And this has always led all the scientific researchers, pharmaceutical scientists, practitioners, clinicians to understand the disease mechanism. I think although we are not doctors, but as pharmaceutical scientists, what disease is and how is it caused, and what are the mechanism of actions of a drug to treat it, and how the pathogenesis of disease progresses. This has all interested us right from the beginning. And Hippocrates very well said that it is more important to know what sort of person has a disease than to know what sort of a disease a person has. So although we had been trying to understand the human biology since long, but unfortunately, as a pharmaceutical researcher, we have not been able to cater to the specific needs of the human biology. I will very quickly give you an example here that most of the time people are nowadays attracted to Ayurveda and Unani. They usually propose that their pathies are actually treating the disease and not the symptoms. However, on our side, where we are dealing with allopathy, we are actually treating the disease. A disease happens, some symptoms come into the surface, and we try to give a medication which will either reduce the inflammation or fever or hypertension. Then we forget about that what exactly the disease is being caused out. But on the other hand, these pathies, which I just mentioned, they are looking forward to going into the deep down and trying to have a personalized medication for that individual based on his temperament, based on the type of lifestyle which he has, and so on and so forth. So this gap has always been there. But the modern medicine has always realized that this gap needs to be filled. So we have always been, right from the inception of the pharmaceutical sciences, we have always been very intrigued and curious to know that how this gap can be filled. So our journey actually started from the very traditional system. We could not actually get to the personalized medicine right in the beginning. So our journey, which is like a few hundred years old, started from what we refer to as your traditional dosage form delivery. And this global pharmacotherapy primarily was giving the best possible sort of formulations, which were low cost, which were highly easy to be manufactured, and which had got clearance from the regulatory very easily. So we can very well relate to this picture where we started with tablets, capsules, powders, oral solution, injectables. And all these formulations primarily were doing their job. They were primarily, and it is not so that they were not helping us to gain health back. They were treating the symptoms and the patient was feeling a bit healthy. but unfortunately it never the disease used to come, we used to consume it, and there used to be some relief. There has always been certain key limitations, and these key limitations were that the chemistry of that molecule was always a challenge. Sometimes there was a poor bioavailability, sometimes the body fat or the tissue will not allow the proper distribution. There would be some issues with the plasma concentration. Sometimes the drug itself will have a short half-life, and many a times if the disease is, for example, CNS disease, the drug will not be able to cross over to the blood-brain barrier and not do what it should do. And there could be a situation that their one size will not fit all the dosing. So there were certain things which were having a problem with this conventional systems. So over the last few decades, we were moving a little closer to what we now refer to as the personalized medicine. Although I will say a disclaimer here that this claim is not 100% true, but the efforts were moving towards the direction of getting the personalized medicine. And these efforts were through something which we now refer to as the nanomedicine. These are some of the scientists who had actually, they are basically physicists who primarily gave the idea of nanotechnology. Then the smart people in the pharmaceutical sector successfully married this idea of nanotechnology and created something referred to as your nanomedicine. So these nanomedicine primarily were small sized, they were designed in a manner that they would directly reach to the target like Ramban, and they will hit the target, and it is more or less very close to something referred to as your personalized medication. The other example which comes to our mind of the commonest personalized medicine is your insulin injection. That the patients who are of a particular age, particular weight, obesity, they will have different units of, and the type of the postprandial sugar or the fasting sugar, the doctor advises that this much unit is being given to this patient. So that is something, the closest example which we see of the precision medicine, although it is 100% not but the other types of implantable insulin devices are there where the blood sugar is immediately tested and immediately the amount of this drug is or the insulin is put into the blood and the patient is getting a very personalized care. So this nano formulation, this insulin devices and all were some of the ways and means by which the pharmaceutical researchers were trying to get closer to the concept of the personalized medicines. This slide primarily is again something which the pharmaceutical researchers are very familiar with. It just gives you an idea about the various types of nano systems which are being available, the advantages which they are giving, and how they are or circumventing the problems of the conventional drug delivery systems, reducing the drug resistance, for example, increasing the drug targeting, increasing the patient compliance, and bringing the formulation closer to the concept of the Ramban or the precision medicine as per the requirement of the patient. But then there is a regret which is there, that despite so much of efforts creating this nano formulation, thinking that, and we have done a lot of research, you will find hundreds and thousands of research papers in the last few years or last couple of decades talking about the nano formulations and doing wonders. But then this one of the articles which we wrote back early 2011 and rather 12 and 13, it's a old article, and where we actually wanted to find out that what could be the reason that these nano delivery systems which have been so supposedly very systematic and personalized are still not able to deliver the basic outcomes. Then we found out that there are certain things which are limiting it, and however hard we try, the behavior of that patient, the tumor, for example, this was basically on cancers. So the tumor primarily was posing certain very specificities which were not common in all the patients. So that were precisely the reason that the outcomes of this cancer therapeutics were not very encouraging. And now we understand that this problems was arising from the nature of the tumor. That means every cancer patient presented a different type of tumor. It was not necessarily the same way it would be treated. It will have biological barriers. It will primarily have different types of convoluted lymphatic drainage. It will have a microenvironment. So in one patient, the tumor was different. In the other patient, the tumor was different. This is precisely the reason that when we do our research in PhDs and other projects, we find very encouraging results in the 2D cell line structures. But as we move from the cell lines to the animal model and maybe to the clinical model, we start seeing the failure of this formulation. So there is a big question which arises, and we should be able to understand and answer this question, that there must be certain inter-subject variability. The tumor presentations is not same in the all animals. It cannot be the same in all human beings. Therefore, there needs to be something which is precisely designed for that particular individual. So we can consider this as the birthright of a system which will give a medication, a right time, and a right dosage form to a right individual in the right measures. Then only we will get the concept of the precision medicines. So this whole background which I have said is, this primarily is just helping us to understand that nowadays the drug delivery, just by virtue of our very hard work and the research which is ongoing, we are trying to move it from systemic to, and passive, to localized, control, and guided. And we are trying to actually do the just hit the sort of, I would say, the magic bullet concept as close as possible. And primarily we are in a position to make some, we are trying to make certain delivery vehicles which will deliver the right drug at the right point at the right patient. And we have to continuously ask this question. We should not rest in peace. Okay, हमने बना दिया, वो वहां चला जाएगा और वहां पर हिट करेगा और फिर जाहिर सी बात है वो बीमारी ठीक हो जाएगी या ट्यूमर श्रिंक हो जाएगा. हमारा जो सवाल यहाँ पर बनता है वो कंटिन्यूसली एक सवाल आते रहना चाहिए कि इज दिस द राइट ड्रग, is this at the right dose, is this for the particular patient, is the patient exactly primarily suitable for this drug. So we are not doing the other way. अब तक हम क्या कर रहे हैं कि is the drug suitable for the patient. अब हम क्या क्वेश्चन कर रहे हैं, is the patient suitable for taking this drug. So this becomes the central argument for the precision medicine. अब प्रिसिजन मेडिसिन के थोड़े और करीब जाने के लिए हम लोगों ने गाइडेड ड्रग डिलीवरी भी कर लिया. अब हम क्या करते हैं कि हमने ट्यूमर को आइडेंटिफाई कर लिया, ट्यूमर हमारा फॉर एग्जांपल सॉलिड ट्यूमर है, एंड वी हैव ट्राई टू मेक अ फॉर्मुलेशन ऑन दैट, एंड देन वी ट्राई टू पुट इट थ्रू द आईवी रूट, एंड बाय यूजिंग अ मैग्नेटिक फील्ड और अल्ट्रासाउंड और एक्सटर्नल स्टिमुलाई और ट्रिगरिंग और whatever route, we are finally delivering that drug to precisely that solid tumor, and we are very happy and sure that now the drug has reached to the right state. But despite even doing this, still the outcomes are not as good as it should be, because we have missed that point that this tumor will not be same in all patients. This tumor primarily will have different epigenesis. This tumor will be associated with different types of inter-subject variability or patient variability. The day I realize that what is this type of tumor and what type of I would say the ligands I will attach, then I will be in a better position to give a precise medication. But we are trying, and from moving from a conventional drug delivery system to a nanoformulation to a highly guided drug delivery system, we are inching closer and closer to the concept of the magic bullet. Now the question comes कि मैजिक बुलेट या प्रिसिशन मेडिसिन का सवाल कहाँ से पैदा हुआ. ऐसा क्या हो गया नॉर्मल रूटीन वाली रिसर्च में, क्या हो गया, क्या गलतियां हो गई कि हमें एक प्रिसिशन मॉडल की तलाश हो गई या एक प्रिसिशन मेडिकेशन करने की जरूरत पड़ गई. तो इसको थोड़ा समझने की कोशिश करते हैं. We are all individuals, we are all homo sapiens, but we are genetically predisposed to certain diseases. हम में से कुछ बहुत एक्टिव मेटाबोलाइजर्स हैं, कुछ हमारे पुअर मेटाबोलाइजर्स हैं, क्योंकि हमारे जो साइटोक्रोम पोर पी, liver is the organ which is the factory of the body and which is undertaking all the processes of metabolism. So we have this cytochrome P450 enzymes, which usually has a role of breaking down the medications. So in a natural, in a normal, healthy, normal human being, the normal enzyme activity is one different, but there can be two genetically different types of human beings. One category can be an active metabolizer and the ultra rapid metabolizer, and the other individual can be a poor metabolizer. सेम ड्रग दी जा रही है, एक की बॉडी में वो बहुत जल्दी से मेटाबोलाइज हो जाती है और दूसरे की बॉडी में वो बहुत स्लो मेटाबोलाइज होती है, बहुत अंतराल तक, बहुत समय तक रहती है. दोनों के आउटकम्स किसी भी हाल सेम नहीं हो सकते. अगर हमने ये समझ लिया कि ये इंडिविजुअल रैपिड मेटाबोलाइजर है तो उसकी जो डोजिंग है वो हमारी अलग हो जाएंगी। हाफ लाइफ की डिपेंडेंसी के बेसिस पे हम उसकी फॉर्मुलेशन डोसेज रेजिमेंट को अलग डिज़ाइन करेंगे और उसके लिए साइड इफेक्ट्स या उसके एडवर्स इवेंट्स को हम कंट्रोल कर पाएंगे। तो ये जेनेटिक वेरिएशन अब तक हमको बहुत ज़्यादा पता नहीं था, लेकिन पिछले चंद सालों में, चंद डेकेड्स में ये जेनेटिक वेरिएशन का हमको काफ़ी ज्ञान हो गया है। एंड हेयर इज वेयर वी आर प्राइमरीली ट्राइंग टू find out a specific tailor made medicine for an individual। दूसरा एक सबसे बड़ा जो गंभीर स्थिति आ जाती है वो आ जाती है डिजीज की हेट्रोजेनिटी में। अब ऊपरी सहित सतही तौर पे वो ब्रेस्ट कैंसर कहलाता है। अब ब्रेस्ट कैंसर है, लेकिन ब्रेस्ट कैंसर मान लीजिए दो मरीज हैं, दोनों के स्टेज 2 ब्रेस्ट कैंसर है। एक दोनों ही स्टेज 2 है, जैसे ही डॉक्टर ने देखा स्टेज 2 है वो इमिडिएटली अपनी कीमोथेरेपी वहां स्टार्ट करने की कोशिश करेंगे। वो कोशिश करेंगे, लेकिन आउटकम क्या होता है कि एक पेशेंट को तो फर्क पड़ा, दूसरे पेशेंट को उसमें कुछ फर्क ही नहीं पड़ा। अब रीजन क्या होता है कि ये ब्रेस्ट कैंसर सतही तौर पे चाहे स्टेज 2 दिखाई दे रहा है, लेकिन अगर हम इसकी गहराई में उतरेंगे तो हमारा एक जो ट्यूमर है, टाइप है, वो हमारा HER2 पॉजिटिव है और दूसरा ट्यूमर टाइप हमारा मे बी एस्ट्रोजन ER पॉजिटिव है। दोनों का ही ट्रीटमेंट हमारा अलग तरह से चलेगा। फॉर एग्जाम्पल, अगर HER2 पॉजिटिव है तो हम हरसेप्टिन या डिफरेंट हमारे मोनोक्लोनल एंटीबॉडीज इफेक्टिव होंगे। ईस्ट्रोजन रिसेप्टर वाले मेडिकेशन में शायद हम टेमोक्सिफिन या टोपिटिकॉन या ऐसी कोई ड्रग के लिए चूज करेंगे। The point which we need to understand is that the breast cancer was same. Ideally, it was a solid tumor, like I showed in the diagram also. But unfortunately, it was not found out whether the one is estrogen positive or the Herceptin positive, and we were giving a common drug to both the patients. One was responding, the other was not responding, depending on whether the mechanism of action was suiting that type of breast cancer or not. The staging is not enough. The conclusion is that just giving a stage 2 designation is not enough. We have to go in for the molecular level, and it should be beyond the physical assessment of that particular tumor. तीसरा एक जो बड़ा कारण होता है जिससे हमारा एवरेज मॉडल या सबके लिए एक जैसा ड्रग हमारा फेलियर हो गया है, वो कहलाता है क्लिनिकल ट्रायल एक्सक्लूजन बायस। And this, what does it mean? When I do a clinical trial, I think all of us are colleagues and we understand this very easily, that when we are talking about the clinical trials, these clinical trials are done right from the beginning. पहले 200 पेशेंट्स में, फिर मैक्सिमम 20,000 पेशेंट्स में या 40,000 पेशेंट्स में आप क्लिनिकल ट्रायल कर देते हो। अब जो क्लिनिकल ट्रायल का जो सब्जेक्ट्स हैं उनके अंदर नॉर्मली वो ज़रूरी नहीं है कि सारी कोमॉर्बिडिटीज़ वहाँ पर मौजूद हों। क्लासिकल केस में क्या होता है कि जब एक्चुअली मरीज अस्पताल पहुंचता है तो ना सिर्फ उसको एक बीमारी होती है, बल्कि उसके साथ बहुत सारी कोमॉर्बिडिटीज होती हैं। क्लिनिकल ट्रायल्स में वो कोमॉर्बिडिटीज वाले पेशेंट्स की जो प्रेजेंस है वो जरूरी नहीं है कि बहुत हो, और जो ड्रग का उस वक्त आउटकम है वो जरूरी नहीं है कि वो उस वक्त सही आए। What do I mean by this? I'll very quickly give you an example here. In COVID-19, most of the patients who were having morbidities like hypertension and diabetes and asthma and COPD, they were more susceptible and they died in large numbers as compared to the people who were not having any of these comorbidities. So the test of the clinical trials has to be done while the real life patient has to be there. So in reality, when a patient goes to a clinic, the patient will be old patient. He will primarily have 2 or 3 comorbidities like your hypertension or your diabetes or your kidney function. And sometimes the patient is taking more than 3, 4, 5 types of medicines, or he is taking polypharmacy. So in that situation, the real behavior of the drug has to be taken into consideration. Clinical trial कभी भी ऐसे मरीजों पे नहीं हुआ था जो कि ओल्ड पेशेंट हैं या जो कि 4 या 5 बीमारियों के साथ ग्रसित है। क्लिनिकल ट्रायल में भी सिर्फ एक मरीज के साथ, एक मर्ज के साथ हुआ। तो वो एक्सक्लूजन बायस जो हमेशा रहता है, that actually makes it a failure, that the drug which was average, made for average patient might not work for all the patients. तो इन सब को मद्देनजर रखते हुए इन सब का जो सोल्यूशन है वो सोल्यूशन है कि हम जो इंटर इंडिविजुअल वेरिएबिलिटी है उसको समझ के अपने मरीज का इलाज करें। सो हेयर कम्स द कॉन्सेप्ट ऑफ द इंटर इंडिविजुअल। अब जो हम डिस्कस कर रहे थे कि वेरिएबिलिटी है, अब वो वेरिएबिलिटी किस किस तरह की है? Inter subject, inter individual variability. First is that There is a polymorphism in genetics. There is a drug metabolizing enzymes, which I was just giving you an example. There are different sets of enzymes. In one person, CYP2D6 is present. In some other, CY2619 is present. These enzymes are in low concentrations, some in high concentrations. Some are having different types of transporters. Some have drug targets which will alter the efficacy and toxicity. So the standard dose of the same drug can be therapeutic in one patient and ineffective in another, and in the third case it can be toxic. So the genetic predisposition and the presence of certain enzymes will let the drug behave differently in the three categories of patient. एक में वो बेहतरीन इलाज कर रही है, दूसरे में कतई इलाज नहीं कर रही है, तीसरे में इलाज तो कर नहीं रही, उसके साथ साथ टॉक्सिसिटी भी दे रही है. वो ड्रग की गलती नहीं है, वो पेशेंट वेरिएबिलिटी की वजह से हो रहा है. अगर हम इस वेरिएबिलिटी को समझ लें और हर बंदे के लिए अलग तरह की मेडिसिन डिजाइन कर लें, सो तो हमारे थेरेप्यूटिक आउटकम्स कहीं कहीं बेहतर हो जाएंगे. फिर दूसरा एक जो वेरिएबिलिटी होती है हम लोग के अंदर, वो जिनोमिक्स वेरिएबिलिटी होती है. एग्जाम्पल है हमारा, जैसे फॉर एग्जाम्पल ट्यूमर हैज डिफरेंट म्यूटेशनल प्रोफाइल्स, जो भी हम आपको एग्जाम्पल बता रहे थे कि कोई किसी के ऊपर EGFR ओवर एक्सप्रेशन होता है, कहीं पर HER2 ओवर एक्सप्रेशन होता है, कहीं पर BRCA ड्रिवन एक्सप्रेशन होते हैं. सो देयर विल बी डिफरेंट टारगेटेड टाइप्स ऑफ थेरेपीज फॉर डिफरेंट टाइप्स ऑफ दीज पेशेंट्स. सो दिस एक्चुअली हैज लेड टू समथिंग विच इज रेफर्ड टू एज योर प्रिसिशन ऑन्कोलॉजी. आजकल जब कैंसर पेशेंट्स का इलाज होता है अस्पताल में, आई थिंक बहुत सालों से, एवरी पेशेंट हैज गॉट अ डेडिकेटेड ऑन्को बोर्ड. तो उस ऑन्को बोर्ड में वो सारे डॉक्टर्स होते हैं, कीमोथेरेपिस्ट जो ऑन्कोलॉजिस्ट होते हैं, ऑन्को सर्जन होता है, रेडिएशन ऑन्कोलॉजिस्ट होते हैं. दे ऑल सिट टुगेदर एंड दे डिस्कस दैट व्हाट इज बेस्ट फॉर दिस इंडिविजुअल पेशेंट. अनफॉर्चुनेटली इंडिया में कुछ बड़े अस्पतालों में वो जेनेटिक और जीनोमिक्स का भी सारी स्टडी करते हैं, लेकिन अक्सर करके नहीं होती है. दैट इज वाई despite whatever the efforts, the chemotherapy fails in some of the patients. But if we know the genetic and genomic of that particular patient and then we decide about the chemotherapy, the outcomes of oncology will be much much better. तीसरा जो वेरिएबिलिटी होती है वो एपिजेनेटिक्स की होती है. These epigenetics primarily are some changes which happen in and around the genetic material. The basic genetic mechanism या जेनेटिक प्रोफाइलिंग बदलती नहीं है, बट उसके आसपास कुछ चीजें हो जाते हैं, जैसे DNA मिथाइलेशन हो जाएगा, हिस्टोन मॉडिफिकेशन हो जाएगा, RNA कोड नॉन कोडिंग RNA रेगुलेशन में फर्क पड़ेगा. और किन किन चीजों से फर्क पड़ता है इन एपिजेनेटिक फैक्टर्स में? बाय एज, बाय डाइट, बाय स्ट्रेस एंड बाय द एक्सपोजर हिस्ट्री. एंड दिस एपिजेनेटिक्स जो होते हैं वो नॉर्मल जो DNA है उसका बेसिक मैकेनिज्म को नहीं बदलते, बेसिक लेआउट को नहीं बदलते हैं, लेकिन दे आर एबल टू डू सम जेनेटिक जीन स्विचिंग ऑफ एंड ऑन. दिस स्विचिंग ऑफ जीन कैन maybe deteriorate the disease, sometimes improve the disease. So these epigenetics is again a variability which will be different in you and different in others, different in me. So one has to understand these epigenetics better in order to give a better formulation or therapeutic outcome. तीसरा चीज है physiology. We all understand that with age, the organ function, be it renal or hepatic clearance, they usually take a toll. They reduce. बॉडी कॉम्पोजिशन फर्क हो जाता है, वजन बढ़ जाता है, आपका फैट डिस्ट्रीब्यूशन अलग हो जाता है, उसके साथ साथ आपको कोमॉर्बिडिटीज हो जाती हैं, जैसे कि हाइपरटेंशन हो गया, डायबिटीज हो गया. इन सबको मद्देनजर रखते हुए हमको समझने की जरूरत है कि ड्रग की फार्माकोकिनेटिक्स और फार्माकोडायनामिक्स में अंतर आएगा अगर हम फिजियोलॉजी को समझते हुए ड्रग को प्रिस्क्राइब करें. इसलिए जेरियाट्रिक मेडिसिन, पेडियाट्रिक मेडिसिन बड़े बड़े अस्पतालों में अलग है. ओल्ड पीपल आर नॉट ट्रीटेड द वे अ यंग adult would be treated. This is precisely the reason because their KFT, उनका LFT, उनके बाकी और जो मसल जो पावर है, मसल का जो रोल है, फॉर एग्जांपल डायबिटीज में, वो सारी चीजें कॉम्प्रोमाइज हो जाती हैं. सो द कंक्लूजन ऑफ दिस डिस्कशन इज दैट वी हैव टू ensure that how the genetics, genomics, epigenetics, physiology, and even the environmental and lifestyle factors are impacting a human being. Based on this, for example, there is a smoker and a non-smoker. There is a person who is having a good gut health, the other is not having a good gut health. There is a person who is belonging to a particular category in a geography where there are high chance of him contracting a disease, where the other person is living in a very good locality. So all these factors, including the disease heterogeneity, which I just give an example by the breast cancer, all these factors are reasons to understand that every individual will not respond in the same manner to a particular drug. One glove does not fit all. एक ही बाण से सबकी बीमारी को नहीं खत्म किया जा सकता. So this basically becomes, this understanding has erupted in a huge amount in the few last years, last decades, and now we are actually understanding it better, and we are moving from the average formulations or average drug therapy to something referred to as your precision medicine. So इस सबका जो discussion का क्या लब्बो-लुबाब मिला, what is the final sort of conclusion of whatever we have discussed in this background is that there has to be a holistic system level approach. We have to understand the precision medicine in terms with multiomics. We should understand the genomics, the transcriptomics, proteomics, metabolomics, clinical history and environmental context to guide the diagnosis and therapy of an individual, rather than isolation, rather than just treating that for that person with the average formulation which we give out in the market. अभी हमारे genomics अभी हमने बता दिया, the sum total of the genomes. Transcriptomics actually is the RNA and the type of RNAs which are active at that particular point when they are translating it into the protein. And proteomics primarily is the sum total of the protein synthesis which is happening. Nowadays we say that disease is actually a protein. What do I mean by this? That every disease has got certain specific protein markers. So कुछ भी ऐसा कुछ बीमारी का आप अगर देखोगे तो you will have certain protein. आप कभी बड़े अस्पतालों की अगर रिपोर्ट पढ़ते हो तो C प्रोटीन बढ़ गया, this protein in the urine has increased, or urinary आपका NFT वो I'm sorry kidney में ये protein बढ़ा हुआ आया. So ultimately every disease is now being expressed in the form of proteomics. So if I understand the proteomics, then metabolomics are actually the presence of the small molecules like your glucose, like your other fat molecules, which primarily are there in the metabolic diseases like diabetes. So a metabolism or the metabolomics of a healthy individual cannot be the same as compared to a diabetic individual. So a person X who is not diabetic is suffering from hypertension and another disease, and the person Y who is already having diabetes, whose metabolomics will be different, will not respond in the same manner to the same drug. So this background has to be kept in mind while we proceed further to understand what is the personalized medication or the precision medicine. Needless to mention, when we are primarily taking this holistic understanding and understanding the intersubject variability, तो बहुत logical है ये बात कि जो हमारी personalized medication है वो बेहतर होगी बाकी routine medication से. Improved efficacy होगी क्योंकि हम matching therapy कर रहे हैं patient की molecular profiling से. We are increasing the response rates compared to the trial and error prescribing. पहले doctors with due respect to them एक मेडिसिन दिया, फिर आप 2 हफ्ते के बाद गए, इससे फर्क नहीं पड़ा, दूसरी मेडिसिन दे दी, फिर आप गए नहीं अभी थोड़ा सा कम फर्क पड़ा है तो या तो उसने डॉक्टर बदल दिया पेशेंट ने, या फिर डॉक्टर ने फिर अगली बार फिर दोबारा से मेडिकेशन बदला. इसका मतलब वो हिट एंड ट्रायल प्रेस्क्रिप्शन कर रहा है. लेकिन डे 1 में अगर वो सारा हिस्टोरिकल डेटा उनके पास आ जाए कि ये मरीज का ये वेरिएबिलिटी है, इसके अंदर ये ससेप्टेबिलिटी है, ये स्मोकर है. आजकल तो डॉक्टर्स, पहले टाइम में डॉक्टर्स 1-1, 2-2 घंटे तक, even today I remember when we used to go in our times, the doctor used to spend at least 1 hour in taking a history. Nowadays they are just doing it in 5 minutes. So they are not getting into the depth of it, that what exactly could be the situation of that individual. Molecular profiling तो खैर अभी आ गई है latest, उसके पहले तो at least history आप ले लो ठीक से कि what are the things which this person is. On the other hand, I have observed as in my younger days also while I was a child, that whenever we used to go to this Hakim and Vaid, they even used to ask you what did you eat last night. They will ask you when did you take the last bath. They will even ask you things which you at that point of time they looked very funny. What is the color of your urine? What is the color of your phlegm or your sputum? But exactly now when we are talking about, I was just making this presentation, so all these things came rushing back to me, that actually they were trying to find out the variability and precisely that what is a problem with this individual with whom we are treating. The today's doctor treat the scans, but actually if you just read any MRI scan, उसमें नीचे एक line लिखी होती है clinically correlate. So understanding the patient is the key, then only we will get an improved efficacy. So that becomes logical step for the personalized medication. The second thing which the personalized medication will do is that definitely it will help reduce the adverse drug reactions. A typical example is that of warfarin, which is a blood thinning drug, and clopidogrel, which is also of the same category. In these cases, some of the people are they are different variable metabolizers. So वारफरिन एक मरीज ने लिया जिसका मेटाबोलाइजिंग रेट बहुत ज्यादा है, जैसे वो हुआ तो उसके अंदर सेम डोज काम नहीं करेगी, उसको हाई डोज देनी पड़ेगी in order to go in for the blood thinning. On the other hand, there is another patient who is a slow metabolizer of warfarin. उसका मतलब क्या हुआ कि ड्रग चली गई, वो हौले हौले मेटाबोलाइज हो रही है, वारफरिन की मात्रा बहुत अधिक समय तक रहेगी और खुदा ना खास्ता उस बंदे का कोई एक्सीडेंट हो जाए या ब्लड लॉस हो जाए, so it will be dangerous for that patient because warfarin was in high quantity and blood clotting नहीं हो पाएगी. So if before I prescribe warfarin as a doctor, if I do a small thing that I go in for genetic profiling and I find out that whether the patient is a quick metabolizer or a slow metabolizer of warfarin, then what extent of warfarin I will prescribe will matter to that person and save the individual from dangerous situations. The other thing, which again a personalized medication through nano carriers and all, what we are trying to do is that we are trying to make a nano delivery system which will guide me to that particular variable tumor, and into this I am then delivering my exact amount of drug as per the overexpression and underexpression of the proteins in this tumor. This is again referred to as theranostics. So ये जो theranostics हो गए या फिर हमारी guided delivery हो गई based on the variability of the individual are all the outcomes of the logical outcomes of the personalized medication. Then the last thing which I would like to discuss here is that personalized medication is actually being preventive and predictive. One example which comes rushing back to me is that of Angelina Jolie, the Hollywood actress. This particular woman underwent a radical double mastectomy. She removed both her breasts. The reason was that the BRCA1 gene, when she went in for genetic profiling, the BRCA1 gene, which is responsible for this breast cancer, had 87% chances that she may suffer from this cancer. So because that gene was primarily working on the breast tissue only, so she just removed both the breast and underwent an implantation of a false breast. And that was one of the ways by which this genetic profiling or pharmacogenomics came handy to this female, and she was able to, at least by God's grace, she is doing well. She has still not reported any of these adverse things because the 87% chances of getting a breast cancer were removed by simply removing the breast itself. Of course, this is not a routine thing for people this side of the world. We will not even have that type of $10,000, $1 lakh in order to get the pharmacogenomic profiling. But nonetheless, this is an example where personalized medications, preventive and predictive care is being proven beyond doubt. That personalized medication or precision medication can help you to prevent a disease by knowing beforehand that at this age, because of the predilection of these types of genes, you may fall prey to a cancer. Then this is exactly the slide. After this background, I think we will all agree that the idea of personalized medication, which customizes treatment to a single patient according to their unique taste, has been a long-standing aspiration in the healthcare sector. Right from the day one, we were all working towards getting this concept. जैसे जैसे मरीज की जरूरत है वैसे ही उसकी customized interventions बन जाएं individual characteristics के basis पे, and then we are primarily looking forward to. So we are already in that scenario where the theoretical framework has already evolved, and we are looking into practical methodology and trying to do some amount of clinical decision making across the specialties and getting as close as possible. Maybe 10 years down the line, 20 years down the line, we may enter into the hospital, and the hospital will just maybe ask us to give us one card, and based on that data which we are having, they will say, okay, this person is suffering from this disease, and primarily this is the therapy which is required for you. We are looking forward to that type of precision medicine. Let's now understand that what exactly has happened in this whole journey. The basic concept of the precision medicine, it is a conceptual shift. So the average patient evidence has now shifted to a concept of this patient interference or inference. So what usually are the things that, as I just mentioned a bit back, that we are having different data which are now available, like the proteomics, genomics, metabolomics, digital health information. We are putting everything together, including the electronic health records, and we are trying to make sure that we are not following that that one size fit all dosing, staging, treatment protocols. Rather, we are looking forward to the exact atypical patient which are not in that guidelines. So normally जो guidelines आपकी इलाज की होती हैं वो डायबिटीज वाला है. बहुत इस बात पे आर्ग्यूमेंट होता है कि कुछ डॉक्टर्स कहते हैं कि 120 फास्टिंग ठीक है, कुछ डॉक्टर्स कहते हैं कि नहीं भैया आपको तो मेटफॉर्मिन पे स्टार्ट करना पड़ेगा. सो इट टोटली डिपेंड्स ऑन द गाइडलाइंस. डायबिटीज में डायबिटीज गाइडलाइंस हैं, किसी और मरीज में किसी और तरह की गाइडलाइंस, हार्ट कार्डियक गाइडलाइंस अलग होती हैं. तो दे आर ऑल फॉर एवरेज पेशेंट्स. But there are certain people who will not fit in these guidelines. Those are referred to as the outliers. And this precision medicine is actually looking forward to treat that atypical patient who is a unique case. So if every individual is treated on the individual base and not treated as an average patient, then the outcomes of these precision medicines will be much, much better. बट देन ऑफ कोर्स वी हैव बीन टॉकिंग अबाउट दैट ऐसे कर सकते हैं, इसका ये फायदे हैं, लेकिन इसके बहुत सारे बैरियर्स हैं, इतना आसान नहीं है. और हमारी तरफ यानी कि दिस साइड ऑफ द वर्ल्ड वेयर वी आर लिविंग, दिस प्रिसिशन मेडिसिन में टेक अराउंड 20 years to come. The basic barriers which are present there, the first the scientific and technical challenge. There is a lot of data complexity, a lot of, as we also presented in our research review article, that there is a lot of translation gap. There are many hurdles, there is a lot of failures. Even I make a precision drug delivery system, उसके बाद में भी बहुत सारे reasons है कि वो काम नहीं करता है. Manufacturing complexity of precision medicine and scale up is again a big challenge. Regulatory and ethic challenge है, जाहिर सी बात है. Data privacy हमको अभी, for example, there are a lot of data privacy rules which have come up. You cannot share the prescription, even you cannot share the history of a patient to anybody. Even sometimes when the court asks, कोई मान लीजिए किसी का डेथ हो गई है और किसी फैमिली ने बोला कि नहीं डेथ में हमें डाउट है कि नेग्लिजेंस हुआ है, तो वो हिस्ट्री भी जो पेशेंट की होगी या उसकी जो प्रोफाइलिंग है, जब तक कोर्ट इंस्ट्रक्शन नहीं देता है डॉक्टर्स को, डॉक्टर्स कैन नॉट शेयर विद द फैमिली मेंबर्स. सो वी हैव गॉट अ ह्यूज अमाउंट ऑफ डेटा privacy concerns. Regulations are not in place. Equity and representation is not there. इकोनॉमिक तो हमने बताया कि ऑलरेडी, जैसे फॉर एग्जांपल जेनेटिक प्रोफाइलिंग में इतने $1,000 लगेंगे. वी डू नॉट हैव दैट टाइप ऑफ sort of arrangement this side of the world. And even if insurance is there, most of the insurance company will not give the reimbursement if I go under. For example, if I decide at this age I want to go in for genetic profiling to find out what disease will I get at the age of 60, and I approach my insurance company कि भाई हमने अपना ये पता करवाया है कि 60 साल की उम्र में हमको क्या बीमारी हो सकती है ताकि हम अभी से बचाव कर लें. Insurance will never pay. You will have to spend lakhs of rupees from your pocket. So that becomes another very important limitation. Clinical workflow integration is still not there. It is not clinician friendly. The doctors, you must all be observing them. They are too busy. They do not have that type of sort of arrangements and support system where they can put their data and translate it and take the help of these all the tools which are available. So it becomes actually very, very, very difficult for the clinical integration. And of course, long-term safety monitoring is important there. If I prepare a gene-based personalized system, today I am making it, I say that today in my laboratory, I am making a personalized medication for treatment of cervix cancer. I am dead sure that including my own self, I will not be putting myself in that test because it has not been tested. It has to be, I don't know whether it will be passing the clinical trial, and pharmacovigilance and delayed adverse reactions can be there. So there is a lot of question marks which are there that agar aapne bana bhi li personalized medication, it would be very difficult to keep a track of the pharmacovigilance and the other related safety aspects. So these are some of the things which we have to bear in mind before we get into the level of AI and personalized medication. So we have understood, I trust. Can I ask you a question here? Am I able to put my points across? जो अब तक बात बोल रहे हैं हम पहुंचा पा रहे हैं अपनी बात?
Yes, ma'am.
Is it clear to all of you?
Yes, ma'am, it's perfectly clear.
Okay, fine. Thank you so much. थैंक यू सो मच. सो द कोर कॉन्सेप्चुअल शिफ्ट की जो बात कर रहे हैं, और अब हमारा अब क्वेश्चन आ गया हर चीज़ में एआई. आज के जो हमारा एफडीपी का टॉक है उसमें भी एआई की बात हो रही है. अनफॉर्चुनेटली और फॉर्चुनेटली इन 2026 वी कैन नॉट गेट अवे फ्रॉम दिस. The only thing is that if I understand the basics, then only I can adopt the AI. If I know what I will do without AI, then what I will do with AI, then only I can integrate it. अगर हमें basics ही नहीं पता है तो we cannot just rote learn सॉफ्टवेयर की इसमें ये डाटा डाल दो, पता होना चाहिए ना कि वो डेटा क्यों डाल रहे हैं हम. So there is a conceptual shift which we should, as teachers also and as practitioners also, we should understand, and then we can actually take help of the AI which is available to us. Up there are standardized guidelines which I was telling you, and that is only for typical patients. But for atypical patients, there will be lot amount of biological differences. A clinical trial will prove that drugs work on the average. वो कभी ये कमाल नहीं बताते हैं कि हर बंदे के लिए ये ड्रग बनना ही है या सबके ऊपर असर करेगी. वो एक डिस्क्लेमर देते हैं कि भैया it will be work in most of the patients, but there will be some unique genetics, metabolic rates, lifestyle factors will also be there, which primarily have to be borne in mind. So normally, traditionally हम क्या करते हैं, standard dose लिया, it fits an average, patient fail, this fail patient is an atypical patient, precision medicine का सहारा लेते हैं, उस atypical patient के लिए हम tailored protocol बनाते हैं और फिर हम उस individual के लिए खास तौर पे एक precision medicine बनाते हैं. ये रास्ता हम लोग अपनाते हैं हर बंदे का इलाज करने के लिए. But 99% we are dwelling on the average concept, and we are making formulations for all types of averages, and we are still not getting deeper into understanding that what is that atypical patient जो उस average medicine से ठीक नहीं हो रहा है, उसकी क्या जरूरत है. तो ये और conceptual shift आ गया है, और इसमें हमारा AI बहुत मदद कर रहा है, क्योंकि जो डेटा आपको चाहिए, अभी आपने थोड़ी देर पहले मेंशन किया कि हमारे पास ट्रांसक्रिप्टोमिक्स होना चाहिए, प्रोटियोमिक्स का डेटा होना चाहिए, जीनोमिक डेटा होना चाहिए. कभी हमने अभी स्टडी कराई अपने एक स्टूडेंट के काम के लिए, तो एक जीनोमिक की फाइल जो आई, एक सिंपल जीन की फाइल 65 पेजेस की थी. इट इज अनइमेजिनेबल दैट अ डॉक्टर विल स्पेंड आवर्स टुगेदर टू स्टडी दोज प्रिंट आउट्स और अंडरस्टैंड दैट. सो यहाँ पर हमको जरूरत पड़ती है AI की, जो एक हमारे सॉफ्टवेयर हैं, एल्गोरिथमिक्स बने हुए हैं, इंटेलिजेंट ह्यूमन डेटा को मिमिक कर सकते हैं. दैट इज वाई दिस कॉन्सेप्ट ऑफ पर्सनलाइज मेडिकेशन इज वेरी वेरी सॉर्ट ऑफ ओपन टू अडॉप्शन ऑफ द AI टूल्स. अब AI हमारा जो है, अब we now shift from, I think we have now understand the basics of precision medicine. We will now shift to the next section, that how this AI is actually changing the scenario. अब जो AI की जो सबसे important चीज है, because of the AI is nothing. AI is actually data mining. Huge amount of data is there, and that data is properly arranged and then inferred, and then the data is captured by the doctors. It is understood by them, and they make decisions right away. The decision for which they would manually take months will be made in a few minutes, and the outcomes will be much, much better. That is the basis of adoption of the AI. Ab AI, definitely it will enhance the evidence-based medication. Why? Because it will accelerate the evidence synthesis, it will bridge the evidence to practice gap, and it will shift the population level data towards personalized care by shifting out and by sifting out the data which is available to me. Precision mapping using personalized biomarker data, that data which I was telling you, we will have massive DNA, RNA database, which will result in improving variant detection. Protein folding कैसी है, उस बंदे के अंदर क्या प्रोटीन काम कर रहा है, क्या नहीं कर रहा है, प्रोटीन का एक्सप्रेशन अंडर है कि ओवर है, वो सारी चीजें हमको AI से बहुत जल्दी पता चल सकती हैं. And then based on this, whatever data is available, there will be a clinical judgment will be shifted. The doctors will analyze the data, they will take very quick decisions, they will risk the slow loss of core bedside skills. And this is also a disadvantage कि वो अपनी जो basic skills हैं dependent हो जाएगा AI के ऊपर. But then primarily the judgment will be much better. That is what we are expecting. Now this slide will be a little technical to understand. अब AI का जो है, या फिर एक simple अगर आप AI की बात करो, तो AI basically एक collection of historical data है. कुछ भी नया पैदा नहीं होता. आपका कुछ डेटा है जो बहुत सारे, फॉर एग्जांपल हजारों लाखों मरीजों का डेटा आपने इकट्ठा किया, उस डेटा को आपने फीड कर लिया, और उस फीड को आपने मशीन, मतलब उस मशीन को आपने लर्न कराया, जिसको आप मशीन लर्निंग कहते हैं. And primarily that learning can be two types, supervised learning and unsupervised learning. Let us try to understand with a simple example. Algorithms basically are like child, एक बच्चे की तरह होते हैं. For example, there is a child and you primarily give him an apple or a banana. अब उसको तो ये नहीं पता कि ये जो नया बच्चा जिसने पहली बार देखा है एप्पल या बनाना, उसको तो नहीं पता इनमें से कौन है. तो आप क्या कहते हो, बच्चे को ट्रेन करते हो कि ये जो रेड कलर का दिखता है ना, जो थोड़ा सा क्रंची सा है, तो थोड़ा सर्कल सा है, ये एप्पल है. और जो थोड़ा सा लॉन्गिश सा है, सॉफ्ट सा है, येलो से कलर का है, वो बनाना है। तो जब आप उसको बार बार वो चीज़ दिखाओगे तो आप उसको बता रहे हो, लेबल कर रहे हो अपने डेटा को कि एप्पल है, ये बनाना है। अल्टीमेटली अगर बहुत ही डेटा फीड हो गया बच्चे के दिमाग में, तो कहीं भी बाजार में जाओगे तो जैसे ही वो किसी भी हॉकर के पास एप्पल देखेगा तो कहेगा मम्मा एप्पल। तो उसने पहचान लिया क्योंकि आपने उसको ऑलरेडी ट्रेन कर चुके हो। तो दिस इज रेफर टू एज अ सुपरवाइज्ड लर्निंग। इसी तरह आपका जो एल्गोरिदम्स को आप लेबल कर लेते हो, ऑलरेडी एक डेटा है, फॉर एग्जांपल एक उसने उसके अंदर आपने डेटा डाल दिया कि ये सिम्टम्स हैं एक मरीज के और ये डिजीज है, और उसका आंसर ये है कि अगर उसके अंदर बुखार है, उसके सांस में तकलीफ है, उसको सांस लेने में दर्द हो रहा है, पेन हो रहा है सांस को लेने में, तो उसको ये इट शुड बी निमोनिया। तो जैसे ही कुछ वो दूसरा डॉक्टर डालेगा वो सिम्टम्स अपने मरीज के, तो उसके अंदर वो इमिडिएटली बताएगा भाई इसका आंसर निमोनिया, या तो वो निमोनिया डालेगा तो सही होगा, अगर वो कुछ और प्रेडिक्ट करेगा तो वो उसको मना कर देगा। अब इट कैन बी डन इन टू वेज। एक तो क्लासिफाई हो गया, जैसे फॉर एग्जांपल आपने बहुत सारे उसमें एप्पल और बनाना को क्लासिफाई कर दिया। सो दिस हेल्प्स द डिजीज वेदर दिस डिजीज इज प्रेजेंट और नॉट। फीवर प्लस कफिंग प्लस स्प्यूटम कलर दिस एंड प्राइमरीली पेन व्हाइल कफिंग इज निमोनिया। तो आपने ऑलरेडी इस लेबल को कर लिया, क्लासिफाई कर लिया कि जब इस तरह के सिम्टम्स इस सिस्टम में फेड होंगे तो आप इमिडिएटली उसको क्लासिफाई कैटेगराइज कर दोगे कि ये भाई निमोनिया है। दूसरा एक रिग्रेशन वाला हो गया। रिग्रेशन में क्या है, कॉन्टिन्यूअस वैल्यूज उसके आते रहते हैं कि अभी ये था, अब थोड़ी देर के बाद इसका फॉर एग्जांपल केएफटी डिटेरियोरेट हो जाएगा, केएफटी में थोड़ा सा क्रिएटिनिन बढ़ा आएगा, तो दिस पेशेंट इज गोइंग इनटू द किडनी फेलियर। सो देयर कैन बी वन मॉडल ऑफ क्लासिफिकेशन, वन मॉडल ऑफ रिग्रेशन, बट everything is being labeled. The data is already put. Medical scans or diagnostic labels are already made. You put a fresh data, immediately that will be compared and you will be told that this is supposedly this. अनसुपरवाइज डेटा में थोड़ा सा अलग करके, अनसुपरवाइज डेटा में फॉर एग्जांपल आपने एक बास्केट लिया फ्रूट्स का, उसमें हर तरह के फ्रूट्स हैं, आपने बच्चे के सामने रख दिया, आपने बच्चे को ये नहीं बताया कि ये सेब है, ये केला है, ये आलू बुखारा है, ये एवोकाडो है, ये वो है। लेकिन वो क्या करेगा, अगर आप उसको ऐसे उसको अलाउ कर दो तो मे बी वो क्या कर सकता है कि वो कैटेगराइज करेगा। वो क्या करेगा कि जो सेब जैसे क्रंची हैं, हार्ड है, उनको अलग कर देगा, और जो पल्पी फ्रूट है लाइक बनाना, शेप इनका एलोंगेटेड है, उनको अलग कर देगा। तो जो एल्गोरिदम है या मशीन लर्निंग वाली जो मशीन है, आपका जो कंप्यूटर है, उसने अपने से उसको कैटेगराइज कर लिया। कुछ डेटा को उसने एज अ एप्पल कैटेगराइज कर लिया, कुछ डेटा को उसने बनाना कैटेगराइज कर दिया, आपने उसको नहीं सिखाया। सो दिस इज रेफर टू एज अनसुपरवाइज्ड और अ सेल्फ सुपरवाइज्ड थिंग. This usually is important because sometimes what happens that you put a data about a particular diseases. For example, there are patients who are suffering from sepsis. एंड देर सेप्सिस, एक तो जनरल टर्म हुई सेप्सिस, लेकिन सेप्सिस के अंदर कुछ आपने तो नहीं बताया उसको, लेकिन उसने क्या देखा कि कुछ मरीजों के अंदर सेप्सिस जो एक पर्टिकुलर टाइप का है, तो उसने उस टाइप के सेप्सिस को अलग से एक वो दिखा दिया कि दिस इज अ सब टाइप ऑफ द सेप्सिस एंड विच यू लेटर ऑन टर्म एज एंडोटाइप्स ऑफ द सेप्सिस. सो दिस अनसुपरवाइज्ड और सेल्फ सुपरवाइज्ड डेटा gives a lot of work to the machine, and the machine, even if when you are not labeling it, you just keep on filling data, वो अपने एल्गोरिदम के बेसिस पे अपनी अपनी कैटेगरीज बना के आपको डाल देता है. कई बार आप फॉर एग्जांपल कोई प्रेजेंटेशन बना रहे हो, आपके पास कोई डेटा 3 4 5 तरह का आ गया, आप क्लॉडे में जाओ या किसी भी एआई टूल में जाओ, आप उसको सिंपल एक प्रॉम्प्ट देते हो, कैटेगराइज अकॉर्डिंग टू maybe number of the data which is available or content of the data which is available. आपने उसको नहीं बताया कैसे कैटेगराइज करोगे, लेकिन वो अपने एल्गोरिदम के बेसिस पे उसको अलग अलग कैटेगराइज करके आपको दे देगा, किसी का टेबल बना देगा, किसी का ग्राफ बना देगा. फिर जब वो डेटा आप देखोगे, देन यू प्राइमरीली आर एबल टू हैव अ रेलेवेंस टू दैट. अच्छा, ये डेटा इन्होंने इसका बनाया है, एज वाइज डेटा ग्राफ का बना दिया. अच्छा, ये डेटा इन्होंने इसका ये बनाया है कि बीपी कितने बजे से कितने बजे शूट हुआ, कितने बजे शूट नहीं हुआ. सो दैट सेल्फ थिंग labeling which the machine is doing, it is referred to, because you are not labeling it. This type of data is called as your unsupervised or self-supervised data. Both these types of data are fed in normal clinical practice, and both these data can be used very well used for the cancer. For example, this self-supervised data becomes very handy and useful to find out the type of cancer. आपके पास कैंसर के बहुत सारे स्कैन्स हैं, इमेजेस हैं, डेटा है, लेकिन आपको पता नहीं है कौन से टाइप का कैंसर है। तो ये आपका जो एल्गोरिथमिक डिजाइन होता है, ये आपको थोड़ा सा डेटा सेग्रीगेट करके दिखाएगा कि बाकी तो कॉमन है, इसमें कुछ अलग दिख रहा है। Then as a clinician, you will understand, okay, this is different. Then you go deep down into it, and then maybe you are able to find out that this is Herceptin positive data, or for example, ER data, or EGFR data, whatever type of tumor it is. So this helps us to understand, to unravel the hidden type of information which is present there. These are some of the architecture. So when I talk about the अब ये for example बहुत मुश्किल है as teachers हमको समझना, लेकिन I am also trying to learn, and I think all of you must be also doing it. सीखना ही पड़ेगा हम लोगों को कि ये जो आर्किटेक्चर है या मॉडल्स हैं, ये हर तरह के अलग अलग हैं। जो आपके प्रिसिशन मेडिसिन में या हेल्थ केयर में जो यूज होते हैं, जो एडवांस्ड इवोल्यूशनरी एल्गोरिथम्स हैं, वो 3-4 तरह के होते हैं बेसिकली। सबसे पहला कैटेगरी होता है आपका कॉन्वोल्यूशनल न्यूरल नेटवर्क्स। ये जो न्यूरल नेटवर्क्स वर्क है, ये बिल्कुल वैसे ही है। ये कहाँ से अडॉप्ट हुआ है? हमारे ब्रेन से। अगर आप ब्रेन का कभी स्कैन करोगे या उसका कभी आपने पिक्चर देखा होगा, तो न्यूरॉन्स भरे पड़े हैं, हर न्यूरॉन के बीच में रिलेशनशिप हो रहा है, दैट मीन्स सिग्नलिंग हो रहा है। तो एक डॉट से दूसरी डॉट पे, तीसरी डॉट पे, चौथी डॉट पे आपस में वो एक दूसरे से बात कर रहे हैं। तो ये न्यूरल नेटवर्क वर्ड वहां से लिया गया है, बिकॉज द डेटा पॉइंट्स, द नॉट्स विच आर देयर, दे आर टॉकिंग टू ईच अदर. दिस इज व्हाट एग्जैक्टली व्हाट द एल्गोरिथम इज बीइंग मेड टू डू. अब कॉन्वोल्यूशनल में क्या होता है कि आपका जो जो कॉन्वोल्यूशनल या CNN टाइप वाला है, ये बेसिकली हमारा मेडिकल इमेजिंग डेटा के लिए होता है। अब इसको थोड़ा सा एक बहुत सिंपल एग्जांपल से समझते हैं हम, कि मान लीजिए हमने बिल्ली देखी। अब हम जो बिल्ली को अगर देखते हैं, तो हम एक्चुअली पूरी बिल्ली को कभी नहीं देखते। हमने उसके कान देखे, हमने उसके फर देखे, हमने उसके मस्टैशेस देखे, हमने उसके ग्लोइंग आईज देखी, फिर हमारा ब्रेन उसको प्रोसेस करके थोड़ी देर पे पूरा एक इमेज बना देता है। तो बिल्कुल यही कॉन्सेप्ट है कॉन्वोल्यूशनल न्यूरल नेटवर्क्स का। मिलियंस एंड मिलियंस ऑफ इमेजेस आर फेड इनटू दिस एल्गोरिथम, एंड देन व्हेन अ न्यू एल्गोरिथम, अब जब आपने एक बार बिल्ली देख लिया और आपके दिमाग में ये इमेज बन गई कि बिल्ली ऐसी होती है, तो आप अगर कभी जंगल में निकलते हो, तो आपने बहुत सारे जानवर देखे, लेकिन अचानक से अगर वही पिक्चर, वही जानवर आएगा, तो आप इमिडिएटली कहते हो, अरे ये तो बिल्ली है, ये तो देखो बिल्कुल वैसी है जैसे हमारे घर में, लेकिन इसका रंग थोड़ा अलग है, शायद जंगली बिल्ली इस तरह से होती है, बट यू आर एबल टू रिलेट टू दैट इमेज ऑफ द कैट. So in this case, what is happening? They are primarily putting the millions of image, then you put your image, and immediately it will just compare that and it will give you that, okay, this is what. अब कोविड के टाइम में क्या हुआ? कोविड के टाइम में एक जो लंग्स का एक बड़ा पिक्चर था, जिसको ग्राउंड ग्लास इमेज बोलते थे, कि वो इमेज बाकी किसी मतलब टीबी में नहीं आती है, किसी निमोनिया में नहीं आती है, निमोनिया में पैचेस आ जाते हैं वाइट वाइट, लेकिन एक पिसा हुआ ग्लास या ग्राउंड ग्लास की इमेज सिर्फ कोविड वाले लंग्स में आती थी। तो अभी उस टाइम पे बहुत सारा डेटा क्रिएट हुआ, और अब लोगों ने एल्गोरिदम में उसको डाल दिया है, CNN नेटवर्किंग में उसको डाल दिया है। तो आज के टाइम में वो हजारों लाखों इमेजेस एल्गोरिदम में है, किसी भी फॉर एग्जांपल हॉस्पिटल के डेटा मैनेजमेंट में। तो वो क्या होगा, एक नया मरीज पहुंचा, उसका एक्सरे किया, और उस एक्सरे को भी अपलोड कर दिया, वो फटाफट उसके साथ पैच करता है कि इसके अंदर ग्राउंड ग्लास इमेज है कि नहीं है। इमीडिएटली वो मैच करता है कि अरे इसका तो अंदर ग्राउंड ग्लास इमेज दिख रही है, और जो हमारे पास लाखों डेटा है वो बिल्कुल उसके साथ मैच कर रहा है। 99% दिस पर्सन इज सफरिंग फ्रॉम कोविड. दिस बिकम्स सो फास्ट. बिफोर द रेडियोलॉजिस्ट गिव्स आउट टू द फिजिशियन आएगा, देखेगा, लेकिन अगर रेडियोलॉजिस्ट ही वो डाल देगा उसके अंदर एल्गोरिथम के अंदर, विद इन मिनट्स दैट रेडियोलॉजिस्ट विल एबल टू 100% कंफर्म दैट दिस इंडिविजुअल हु इज एडमिटेड इन द हॉस्पिटल इज सफरिंग फ्रॉम COVID-19 on 26 January 2026. So this becomes so fast. दूसरा एक इनका तरीका क्या है, वो है रिकरेंट न्यूरल नेटवर्क्स. ये बिल्कुल ये क्या यूज करता है, ये टाइम बेस्ड या ये वर्ड्स को पिक करता है. फॉर एग्जाम्पल एक वर्ड है बैंक. अब बैंक के 2 मीनिंग होते हैं. अब इसका जो हम सेंटेंस में यूज करेंगे, जो इसका प्रीसीडिंग वर्ड्स है, उससे आपको समझ आएगा कि हम कौन से वाले बैंक की बात कर रहे हैं. सो हम पहले एक सेंटेंस बोलते हैं, आई वेंट टू द बैंक टू डिपॉजिट मनी. Then I said, say a second sentence, the children were playing near the bank of the river. जो पहले प्रीसीडिंग वर्ड्स थे, उन प्रीसीडिंग वर्ड से ही आप समझ गए हम कौन से वाले बैंक की बात कर रहे हैं। पहले केस में हम उस बैंक की बात कर रहे हैं जहां पर हम पैसा जमा करते हैं, जो नॉर्मल रूटीन हमारा बैंक होता है। दूसरे वाले में हम उस बैंक की बात कर रहे हैं जो कश्ती या सॉरी नदी का किनारा होता है। कहाँ से पता चला? चिल्ड्रन आर प्लेइंग नियर द बैंक ऑफ रिवर। सो ये जो प्रीसीडिंग वर्ड्स हैं, समटाइम्स आप कोई कीबोर्ड में डेटा डालते हो, आपने 2-3 वर्ड्स डाले, ऑटोमेटिकली वो आगे सजेशंस देना शुरू कर देता है। सो दैट इज एग्जैक्टली हाउ दिस रिकरेंट न्यूरल नेटवर्क वर्क्स। फिर आपका दूसरा आ जाता है नेचुरल लैंग्वेज प्रोसेसिंग। नेचुरल लैंग्वेज प्रोसेसिंग का बड़ा टिपिकल एग्जांपल है, आप फोन उठाते हो, आप कहते हो सीरी कॉल डॉक्टर ज़ीनत इकबाल, तो जो आईफोन वाला है वो अपने आप ही उसको नंबर देता है और एंड फाइनली प्राइमरीली वो आगे का काम उसको बता देता है कि सीरी ये कर लीजिए। या फिर आपका चैट बोर्ड्स होते हैं, आपको कोई कपड़ा पसंद आ गया, कोई ड्रेस पसंद आ गया, इमीडिएटली आपके पास मैसेज आ गया, कैन आई हेल्प यू, यू हैड डिसाइडेड दैट यू वांटेड टू बाय दिस एंड सो फोर्थ। तो सिमिलरली इफ यू आर गिविंग दिस प्रॉम्प्ट्स, वी आर, अब जैसे नेचुरल लैंग्वेज में हमारी अपनी लैंग्वेज है, लेकिन कंप्यूटर की लैंग्वेज क्या होती है? पाइथन होती है, 0-1 की बाइनरी कोड होती है। अब हमको वो लैंग्वेज क्यों सीखना है? अब हमने एल्गोरिदम्स को ऐसे बना दिया कि जो हमारी नेचुरल लैंग्वेज है वो उससे मैसेज लेके वो आगे उसको कन्वर्सेशन को कंटिन्यू कर देता है। सेम चीज आप उसमें करते हो चैट जीपीटी में, यू विल गिव अ प्रॉम्प्ट, दे विल गिव यू एन आंसर इन योर लैंग्वेज। Similarly, the other things can be novel representation, knowledge representation from different area, and ultimate goal is that it will drive the advanced patient personalization from preventive care to chronic disease management. So I think I've been able to give you a little feel of how this AI architecture or AI models are being used. These is examples of the same thing which I have discussed in detail. यहाँ पर एक typical model होता है, जैसे random forest है. These are all types of algorithms. So random forest आप समझ सकते हो बिल्कुल plant की तरह या tree की तरह ensemble होता है कि एक data का main edge है, फिर उसकी branches से आपने different information निकाली, फिर आपने उसको correlate करा। जैसे CNNs में है रेडियोलॉजी की इंफॉर्मेशन, पैथोलॉजी की, डर्मेटोलॉजी की, रेटिनल फंडस फोटोज की, ये सारी इंफॉर्मेशन कलेक्ट करके आपको डायग्नोसिस में मदद कर देगा। सीक्वेंशियल डेटा में आपके क्लिनिकल नोड्स, जीनोमिक सीक्वेंसिंग, ऑल दीज थिंग्स विल हेल्प। देन ग्राफ न्यूरल नेटवर्क्स इज अगेन अ वेरी इंटरेस्टिंग थिंग. It is something which will help us to relate the drug with the protein. For example, drug A, B, C, protein XYZ. कौन सी ड्रग कौन से प्रोटीन के साथ इंटरैक्ट करेगी, और कौन सा प्रोटीन कौन से प्रोटीन के साथ आपस में इंटरैक्शन करेगा, कौन सा ड्रग टारगेट के साथ प्रिसाइजली इंटरैक्ट करेगा, ये सारी इंफॉर्मेशन के साथ में वी आर इन अ पोजीशन टू गिव एन इंपॉर्टेंट थिंग. ओके, दिस इज अ ड्रगेबल साइट. At this site the drug will go, it will interact biologically and biochemically, and then it will give me this outcome and the disease will be treated. So all these types of, so आप हम लोग अक्सर करके when I was studying तो I got very overwhelmed that ये क्या क्या चीज़ है, समझ नहीं आ रहा है. But if you go a little deep down into each one of these things, these are actually the tools to just assimilate the knowledge. ऐसा नहीं है कि हम अभी नहीं कर रहे हैं, पर अभी हम फाइल पे फाइल पढ़ते हैं, डेटा का डेटा पढ़ते हैं, कुछ हम मिस कर देते हैं डेटा. And this what they are doing, they're trying to pull all the data together so that a holistic picture of that person comes and the disease comes and the treatment becomes accordingly tailor made and customized accordingly. These are some of the other tools, the white box models and the black box models. They are again very technical. We just have to make, basic thing is that white box models at the name, one of the basic problems with this artificial intelligence is ये काम तो, for example आपने chat GPT में कुछ भी मांगा क्वेश्चन उससे, फॉर एग्जाम्पल किसी भी चीज के बारे में बात कर रहा है उन्होंने, आप चैट जीपीटी से ये पूछोगे हाउ डिड यू रीच इट, इफ यू आस्क द चैट जीपीटी ये आपने कैसे पाया, ये कैसे आपको मिला, chat GPT will not be able to answer. So the black box models are those where a lot of issues are there, where they are not able to answer what is if and then. But in case of white box models, they are able to answer the if and then steps. If the patient is suffering from this, then this will be used. So there are clear question and answer, and such models which are easily auditable, already reliable by the clinicians, are called as white box models. And there can be another situation where the information is available, but the information is not giving me the reason why this has happened. उसने diagnose कर दिया pneumonia, लेकिन reason नहीं बताया कि pneumonia क्यों है. Such model will be a black box model, the one which is going to be giving me the clinical operational benefits, and I am able to audit it, check it validity. That is called as your white box models. अब नाउ द क्वेश्चन कम्स कि जो हमारा ये साइकिल है या जो मशीनरी है एआई की, उसके अंदर क्या क्या रॉ मटेरियल चाहिए, क्या क्या रॉ सबस्ट्रेट चाहिए. तो जो डेटा है अवेलेबल, हमारे पास इलेक्ट्रॉनिक हेल्थ रिकॉर्ड्स होने चाहिए जिनको हम यूज़ करें, जिसके अंदर हमारे डायग्नोसिस, मेडिकेशंस, फिर हमारे क्लिनिकल डेटाज होते हैं जो डॉक्टर्स नोट्स लिखते हैं हर बार. देन वी हैव गॉट अ लॉट ऑफ इमेजिंग, रेडियोलॉजी, डर्मोस्कोपी, रेटिनल फंडस, फिर हमारे पास जिनोमिक, मल्टी ओमिक डेटा होता है, ट्रांसक्रिप्टोमिक्स. देन हमारे पास आजकल वेरिएबल एंड कॉन्टिन्यूस फिजियोलॉजिकल मॉनिटरिंग. आजकल आपके पास वो कॉन्टिन्यूस ग्लूकोज मॉनिटरिंग वाले पैचेस आते हैं कि आपका हर वक्त आपने क्या खाया, क्या पिया, कितना ग्लूकोज का स्पाइक हुआ, वो सारा आपके डॉक्टर्स तक पहुंच जाता है. देन सोशल एनवायरमेंटल डिटर्मिनेंट्स, द लाइफस्टाइल which I am doing, the type of age which I am having, the environmental factors in which I am living, all this data needs to be fed in order to get the best outcomes. Personalized medication primarily, as I have told you in the earlier, this is at the risk of repetition, but very quickly, it can be used in drug development, clinical decision making, predictive analytics, AI techniques, data integration. All these things will be the tools and the steps for the precision medicine. Personal medicine में I think I've already explained this, that we have got different layers of intersubject variability. हमें उनका genome, epigenome, transcriptome, proteome and metabolome और microbiome. This is again very interesting. The gut health, I think most of the time so many YouTube shorts are available where the people, celebrities talking about the gut health. So in today's time, I was reading somewhere that trillions of bacteria are there in our body. As I'm sitting still, 10,000 particles will be emanated, many will be the bacterial and the microbial particle. So इतना ज़्यादा amount में हमारे mouth में 370 species bacteria की, gut में amazing number of bacteria is present there. So we cannot isolate the treatment of a patient without knowing the gut microbiota. So microbiome is also now playing a very important role. That अभी बहुत recently हमने पढ़ा कि अल्जाइमर के डिजीज में बैक्टीरिया इज प्लेइंग एन इंपॉर्टेंट रोल. पेरियोडोंटल बैक्टीरिया जो है अल्जाइमर कॉज कर सकता है, जो आपके माउथ में इन्फेक्शन होती है. सो दैट हैज बीन फाउंड एंड इन द अल्जाइमर ब्रेन्स it has been found that some of the porphyromonas gingivalis bacteria is present there. So that is a type of integration which has happened. So if I'm taking care of all these 5 layers, I will definitely make a system which is going to give me a personalized medication. These are the applications. I think I've already explained it one way or the other. The take-home message is that we cannot do it with a pen and a paper or a file work. We have to take care of certain types of systems like Marvel AI system. There are a lot of programs which are available. So the first concept is that the healthcare professional is trying to give the same medication to all the 3 types of patients. One patient is getting the expected effect, the second is getting no effect, the third is getting poor thing, the adverse effects. In the other case, when I'm using the AI assisted personalized medication, all the 3 people are getting different medication. Just see, the shape is different here. यहाँ पर तीनों का सेम कैप्सूल दे रहे थे, यहाँ पर इसके लिए अलग बना दिया, इनके लिए अलग बना दिया. So everybody is getting the expected effect because we have taken care into all these 5 systems before we prepare this medication for every individual. This is again a slide which is just helping us to understand that all the information which is present there from all the variables, the pathology, the predictive algorithms together will help me to get a person who is going to get the best fitted medication for that particular individual. AI is also used in drug discovery, although it is not the topic of today's presentation, but just a very fleeting slides that we are now using all the things which we have been using for 10 to 12 years, doing the study from pre-discovery to the last large scale. This thing, every step is now being supported by AI, and the movement of the data has become very fast. And you are in a position to get a new molecule in a few years as compared to the decade in the earlier times. This is the evolving landscape. As I was just telling you, we start from the identification and validation of the targets, and now we can use the concept of precision medicine. हम वही targets को देके जो हमारे मरीज के चाहिए. So and then these are some of the steps not related to today's presentation, but I thought that this will be a little intriguing. We are using it in drug discovery. discovery. We are already are aware of these usages. Then treatment optimization, I gave you an example, where resistance of that drug, then the different dose combinations. Just imagine that we are making a combination of these two drugs. We take care about the routes of administration. We are taking care of the ADME profile of that individual, and then we are predicting whether this drug will be giving a positive treatment outcome or a negative treatment outcome. So we have come to this level. In oncology treatment, I have given example earlier. If I understand the cancer type, if we are able to understand what is the tumor profiling, what are the types of radiotherapy required. So all the onco board, they sit together and they decide that what type of treatments are to be required. कीमोथेरेपी कौन सी देनी होगी, रेडिएशन ऑन्कोलॉजी बताएगा कि रेडिएशन इस मरीज के लिए क्या होगी. Then the other doctors, the surgeon will say that surgery will be done before or after. So that is the type of attention which is being given to the oncology precision medicine. This is again very important for the rare disease. Rare disease का diagnosis में बड़े issues होते हैं. Symptoms क्योंकि मरीजों की तादाद इतनी कम होती है उनसे sufferers की, so the data is also not precise. So these algorithms help us to spot complex disease complexes, like for example, detecting the lung cancer nodules in a CT scan, which is not routinely found out, identifying pneumonia, diagnosing diabetic retinopathy from आँख टेस्ट कराने गए, वो डॉक्टर ने बताया या उसने AI असिस्टेंट ने बताया कि भाई आँख का जो आप विजन टेस्ट के लिए आए हो, लेकिन आपका जो डायबिटिक रेटिनोपैथी दिख रही है. उसके पहले इस मरीज को पता ही नहीं था कि मुझे डायबिटीज है. फिर आँख के डॉक्टर ने उसको रेफर किया डायबिटीज के क्लिनिक में, बिकॉज द AI tool has picked up retinal retinopathy, and that is exactly how this has happened in our family, that the patient went to check get the eyesight check. Then the ophthalmologist told him that please go to the diabetologist tomorrow, and the HbA1c was found to be 10, which is a very, very high HbA1c. So these are the types of tools which is helping. CRISPR precision medicine is again a tool which is very much commonly used in your oncology to precisely go in for the CAR T therapy. I think in the interest of the time, I'll have to rush through a few slides. Otherwise, the next part I will not be able to cover. You can just imagine that this you can just see this image, that the lung profile is being seen. And then this data is helping us to match that what is this lung showing. There's a ground glass image here. And then primarily they will match with the ground glass images which is present in the data which has already been fed. Validation is important. I think validation is key to all the types of experimentation. Even if we do it in the laboratory, we validate our data. Similarly, AI tools have got different mechanisms by which the data cannot be generalized. It has to be validated. It has to be the real world. There will be certain algorithmic openness which needs to be put into place. Ethics are already there, which needs to be done. I think I already told about it, that data privacy is one of the biggest hurdle. Then data quality in the EHR, हमारे यहाँ पर unfortunately अभी भी electronic health records नहीं है सारे अस्पतालों में. Incomplete entries होती हैं, डॉक्टर्स के पास टाइम नहीं है कि वो पूरा data upload करें. So all these types can result in, and mostly we have got a huge digital divide. Most of us are not able to use our wearables. We are not able to use our mobiles for the health tracking. So these are some of the limitations. So we have to identify this. The ethical consideration has to be there. All the things has to be done as per the FAIR principles given by the government, and we have to make sure that fairness metrics are followed. These are some of the rules and guidelines which are there, which are present online and at USFDA and the other websites, which helps us that to what extent artificial intelligence can be used and what are the things which we need to keep in our mind. Now the next section, the last section of the presentation, is about the clinical decision support. There's a simple thing that from precision medicine to clinical decision, these are two different things altogether. Clinical decision support is a generic thing where we use a clinical decision in all types of diseases. Let us take an example, that there is a patient who is 65 years. He comes with chest pain. Medical history is type 2 diabetes. He has hypertension. He has a myocardial infarction, and his ECG and blood tests are available. The question asked is, how can clinicians quickly integrate all this information to make the best treatment decision? So what we do, there's an algorithm which is present here. The patient will come, medical history will be taken, all the laboratory results will be integrated, ECG and medical imaging will be sort of captured. Then there will be this AI-powered clinical decision support system which will help to generate evidence-based recommendation. The clinician will review the recommendation and final clinical decision is being made. One has to keep in mind that this clinical decision support system can never replace the clinicians. It only helps them to understand and integrate the data at a higher level, at a quicker level, so that all the things एक घंटा reports लाने में, लगाने में, देखने में लगाएगा. So all these things are being captured very easily in a very short time by the AI algorithms. It usually helps. What are the objectives? It usually helps to improve the diagnostic accuracy. It will recommend appropriate treatments, reduce the medical errors, enhance the patient safety, and improve the healthcare efficiency. And everything is done based on the evidence, patient specific, real time, and the clinician has to be present there. Basically, how it works, patient data has to be captured from the electronic health records. Clinical knowledge base guidelines and research are already there as a part of this algorithm. Clinical data machinery, as I explained earlier, the machine learning has to be done ahead. Recommendations can be given in the form of alerts. भाई अलर्ट देगा, वो बताएगा कि रिस्क हाई है इस पेशेंट की, इसको इमीडिएटली कार्डियक सर्जन के पास जाना चाहिए. डायग्नोसिस बताएगा, ट्रीटमेंट सजेशन बताएगा. उसके बाद जो उसकी रिकमेंडेशंस है अल्गोरिदम की, वो फिजिशियन इवैल्यूएट करेगा. उसके बाद वो फिर उसकी जो पेशेंट केयर होगी, दैट वुड बी डेफिनेटली इम्प्रूव्ड. So this is the historical perspective. We have started from 1970s and now we are moving from up to 2026. So we started with simple simple understanding and collation of data, but now we have come to a level where we are using high. But the only thing which was actually occurring again to me was that it is just an interplay of the data which is available. The more data we are able to process, the better outcomes we are able to give. And all these machinery are just to, so it will be us and the doctors who will help the algorithm to design. So a computer scientist will not be in a position to derive a data, make a data till we are able to tell him. So what is the level of patient information we want to keep? What are the clinical data resources which we will want to take? Data process कैसे होगा, एनालिटिकल इंजन यहाँ पर कैसे काम करेगा, क्या हमारे वो डॉक्टर्स के नोट्स को पढ़ेगा, उनकी भाषा को समझेगा, उसके बाद वो अपनी लैंग्वेज में इसको ट्रांसलेट करेगा. अल्टीमेटली इट हैज टू बी डन बाय द फिजिशियन रिव्यू एंड द बेस्ट पेशेंट आउटकम्स आर गिवन देयर. So these are the technologies. I need not repeat them. Machine learning has to be done, deep learning, NLP, predictive analytics. These are all very technical. How we use them is important. But ultimately, this is an example where we can see that there was a clinical challenge. A patient presents with fever, cough, fatigue, and shortness of breath. Question asked is, what is the most likely diagnosis and what additional investigations are required? Could this be pneumonia, COVID-19, heart failure or pulmonary embolism? वो सारा डेटा कैप्चर करेगा, पेशेंट की डेमोग्राफी, मेडिकल हिस्ट्री, लेबोरेटरी रिजल्ट सबको इंटीग्रेट करेगा, मेडिकल इमेजेस को, क्लिनिकल नोट्स को, अपना एल्गोरिदम रन करेगा. उसके बाद में डिफरेंशियल डायग्नोसिस देगा. उसके बाद में वो डिफरेंशियल डायग्नोसिस जो उसने दिया, फॉर एग्जांपल वो एल्गोरिदम बता रहा है कि 87% चांसेस बैक्टीरियल निमोनिया, वायरल निमोनिया के चांस 8% है, हार्ट फेलियर के 3% है, पल्मोनरी एम्बोलिज्म के 2% है. ये डेटा डॉक्टर देखेगा, उसके बाद डॉक्टर डिसाइड करेगा कि ठीक है कि नहीं, तब वो डायग्नोज करेगा that the patient suffers from bacterial pneumonia. So this, as you know, it is just a support. It can never replace a clinician. Another example on the similar lines, this is an example of drug-drug interactions. So the different types of drugs are being given. बहुत सारे हमारे मरीज जो होते हैं वो सेम, फॉर एग्जांपल ट्राइमेथोप्रिम सल्फामेथोक्सोल के साथ में वारफरिन दे रहे हैं. तो वारफरिन के साथ में अगर ये ड्रग दी जाएगी, then it will increase anticoagulant effect of warfarin. So it gives an alert. So these are something which is beautiful that the algorithms will help us understand, because we keep on, कई बार क्या होते हैं, मरीज अपने प्रिस्क्रिप्शंस को एक डॉक्टर को दिखाएंगे, अगर मानो साइकेट्रिक प्रॉब्लम है और वो न्यूरोसर्जिकल प्रॉब्लम है, तो साइकेट्री वाली प्रिस्क्रिप्शन न्यूरोसर्जन को दिखाएंगे ही नहीं, एक साइकोलॉजिकल इशू रहता है. सो इसमें EHR जब हमारा इलेक्ट्रॉनिक हेल्थ रिकॉर्ड डेटा होगा, उसमें हर तरह की चीज उसके अंदर लिखी होगी. डॉक्टर्स के लिए बहुत आसान होगा कि अब पर्सन हु इज हैविंग पॉलीफार्मेसी, वो ट्राइमेथोप्रिम भी खा रहा है, साथ में वारफरिन पे भी है बेचारा, तो वारफरिन अगर वो दोनों दिया जाएगा तो वारफरिन की वजह से उसका ब्लड लॉस हो सकता है, अगर कट लग गया है, गिर गया. सो वी विल मेक श्योर दैट द डॉक्टर विल चेंज ट्राइमेथोप्रिम एंटीबैक्टीरियल टू सम अदर ड्रग्स. Then these are some of, this is just an example that data हमारा कितना convoluted होता है और हमारा algorithms उसको साफ सुथरा करके it helps the doctors to actually make it into a proper thing. This is some of the pictures which are indicating that how this thing architecture of AI can help you understand the algorithms. That's it. I think this is again something which we have already talked about. तो real world applications हमारा है कि we can actually diagnose the disease very early. We can find out a very nice diagnosis. We will have रेडियोलॉजी में तो बहुत इम्पैक्टफुल है इसका यूज. सो वी कैन गेट द इमेजेस. आपके जो CNN है उसके इमेज को फटाफट वो डॉक्टर्स को बता देते हैं मिलियंस ऑफ इमेज के साथ कंपेयर करके कि द चांसेस आर दैट दिस पर्सन इज सफरिंग फ्रॉम ब्रेन हेमरेज. So all these things can be used for the clinical decision support system. These are the references which we had taken and I deeply acknowledge them. Thank you so much for your kind attention. And I think we all believe that advanced screening, AI technology, and precision therapeutics, every disease can be treatable and preventable, and mortality is no longer inevitable. That is exactly why we are working so hard, and we are trying as pharmaceutical scientists and researcher to try to get as much as possible from the AI and use it intelligently. We cannot afford to use it by simply saying it that we just put it into action. We have to use it very, very intelligently. Thank you so much for your patient listening. I hope and I trust that I have done justice to the topic and I have been able to make you understand some of the aspects. It was very difficult for me also to make this presentation, but thank you so much for giving this opportunity. It was a learning experience for me. I'm open for questions if there are any.
Ma'am, anyone if have any question regarding ma'am session, please share their questions.
Am I audible to you?
Yes, sir.
Yes, you are, please.
Good afternoon, ma'am. Ma'am, I need to know that whenever
Good
we are
afternoon.
developing the algorithm and we are utilizing that in term of analyzing the patient data, what are the platform through which being a pharmacist or young pharmacist we can learn all these things? Like there could be any video kind of thing where we can use and learn it. Because normally, yes.
No, there are a lot of videos now, and unfortunately, most of the data, most of these platforms are paid. Usually, the hospitals are having access to it. हमारे लिए as such, लेकिन I think there are a lot of YouTube presentations which are available. So I will very strongly recommend that if you can just use this Claude, which is an artificial intelligence platform, which will give you at least an insight into the basics of this. अदरवाइज नॉर्मली सारे जो प्लेटफॉर्म्स जो यूज करते हैं ना जो हमने बताया मार्वल और ये बायो अरे ऑल दिस प्राइमरीली आर पेड.
Okay. Thank you very much, ma'am.
Okay.
Thank you very much, ma'am, for such an insightful session. Your enlightening presentation on artificial intelligence in personalized medicine and clinical decision support has provided us with a deeper understanding of how AI is reshaping the future of healthcare through precision medicine, improved diagnostic, and patient-centered clinical care. We sincerely appreciate you for accepting our invitation and taking the time to join us today. Your expertise, inciting thoughts, and valuable guidance have greatly enriched this faculty development program. As a token of our gratitude and appreciation, we are pleased to present your certificate of appreciation and a virtual memento. Ma'am, this is
Thank you so much.
the
I deeply
certificate of
accept it.
appreciation.
I deeply accept it. Thank you so much. And I trust. Honestly, बताइए कुछ समझा पाए, कुछ share कर पाए. Was in a position because
Yes, ma'am. Yes, ma'am.
myself and as a teacher, I was struggling while I was making this presentation because there were so many new things which I had to learn. So, but it was a great learning experience. I hope that I was able to do some justice to the topic. Thank you so much for this opportunity.
Uh, the soft copy of these tokens, ma'am, we
Thank you
will
so
share
much.
you shortly on your email. Once again, thank you so much, ma'am, for your gracious presence, your valuable time,
Okay.
and your outstanding session. Thank you so much, ma'am.
Good afternoon, Vibhu Sawhney sir. कैसे हैं आप?
A very good afternoon to you, Dr. Jinath.
How are you,
Really,
sir?
it was wonderful session, and I'm
Okay,
observing that most of the people are appreciating your this session.
okay,
Thank you very
fine.
much for joining and
Thank
giving
you
us
so
your
much,
precious time.
sir. It means a lot. Thank you so much, sir.
Thank
Thanks
you.
a lot. I'll take your leave
Yeah,
now. I'll
please,
disconnect. Thank you
sir,
so much.
please.
Okay.
Thank you so much, ma'am. With this, we move further. I want to inform that there is a few changes in our schedule. Dr. Sushma Telgaonkar ma'am session will be scheduled for today's second session, but some urgent commitment she is not able to join us today. Dr. Arvinda Pai sir session is exchanged with Dr. Sushma Telgaonkar ma'am. So it's time for welcoming sir for the second technical session of day 4 of faculty development program. It is my great privilege to introduce our distinguished speaker, Dr. Arvind Pai, Associate Professor, Department of Pharmaceutical Chemistry, Manipal College of Pharmaceutical Science, Manipal Academy of Higher Education, Manipal. Sir is a distinguished academician and researcher with more than 18 years of experience in teaching and research. His area of expertise include medicinal chemistry, computer-aided drug design, molecular modeling, molecular dynamics simulations, artificial intelligence assisted drug discovery, and the rational design and synthesis of novel therapeutic agents. His research is focused on the discovery of novel therapeutics for cancer, neurodegenerative disorder, infectious diseases, and metabolic disorders, integrating computational and experimental approaches to advance translational drug discovery. Sir has authored more than 86 research publications in internationally reputed journals and has received competitive research funding from AICTE and the Rajiv Gandhi University of Health Sciences. He also served as a reviewer for leading international publishers including Springer Nature, Elsevier, Bentham, and the Indian Journal of Pharmaceutical Sciences. His current research focuses on integrating artificial intelligence, structure based drug design, molecular simulations, and medicinal chemistry to accelerate the development of next generation therapeutics. Today's session is on the topic AI in pharmacy automation and public health analytics. We are truly honored and delighted to have Dr. Arvinda Pai sir with us today to share his expertise, research experience, and valuable perspective. Hello,
Good
sir.
afternoon. Yeah. Uh, am I audible? My voice?
Yes, sir. Yes, sir.
Okay. So I'll share the presentation. So good afternoon, everyone. So there are people which I may not be knowing. So Vibhu Sahani sir, good afternoon.
Yeah,
Nice to
very
see
good
you, sir.
afternoon. Very good afternoon, Dr.
Yeah,
Pai.
good afternoon,
Thank
sir.
you very much for giving the precious time, and
Yes, sir.
nice to see
Thank,
you after a long time.
yes, sir. Thank you very much, sir.
Great.
So
Wish you all the best for today's session.
thank you, sir. Thank you very much.
Yeah, please proceed.
Yeah. Okay. So can you confirm, please? My slides are visible.
Yes, sir.
Okay. Okay. So very good afternoon, one and all. So thank you for giving me an opportunity for giving a short lecture on AI in pharmacy automation and public health analytics. So even though I'm not directly linked to this public health analytics and this, but I have been working on artificial intelligence based methods from last 10 years. So whatever the softwares and hardware things, they are operating in AI. So it is almost similar to whether it is a drug discovery or whether it is a healthcare automations or industrial automations or any other types. So that's why I feel that this will be a very good platform to share my little knowledge on all these techniques, particularly in pharmacy automations and public health analytics. So I usually focus on how machine learning and automation and predictive analytics are reshaping our pharmacy and public health decision making. So it is now, I think, the new pharmacy NEP syllabus, which was to be implemented from this year, 2026. So a lot of AI-based programmings like Python programming, machine learning techniques have been incorporated. So thank you very much for our PCI team headed by Dr. Montu Bhai Patel sir and his team. They have taken initiative in redesigning the syllabus, and lot of AI-based things have been added in the syllabus, and that is definitely required for our future. So future is based on all this learning of AI techniques and application of these AI techniques in our domain knowledge. So that's why I want to thank again for Pharmacy Council of India and all the member councils. So for introducing the new syllabus with more emphasis on AI-based techniques in the syllabus. So I will start with my presentation. So these may be the contents of my presentations, like what are the foundations of AI? So what is the application of that AI in pharmacy, automation technologies, AI pharma manufacturing, and what is the application in clinical trials? Also in regulatory and quality systems, public health analytics, and there may be some outlook and future discussions. So what are the foundation of the AI? Why it is required? Artificial intelligence, and at present why it is required. So it will be setting a shared definitions and the economic, scientific, and public health pressures, which is pulling AI into pharma and particularly population health. So AI being extensively used now in healthcare and other related pharma aspects. So let us see what are the important AI technologies which we will implement in particular things like machine learning. I think almost all of you have heard about machine learning techniques. So machine learning is nothing but algorithms. So there are some computer algorithms generated. There are programming type. So which learns statistical patterns from the data rather than following a hand-coded rules. So random forest models, gradient boosting for ADMET prediction, which will be most helpful in initial phases of drug design discovery. Deep learning, so multi-layer neural networks that learn hierarchical presentations directly from the raw data. So this will be used in image-based quality control and protein folding. So next one is natural language processing, that is called NLP. So these models that extract structure from unstructured text, literature mining, adverse event reporting, and regulator dossiers. Next one is generative AI and foundation models. So large pre-trained models like diffusion transformers, adaptable to many downstream tasks. So de novo molecule generation and clinical note summarization. So I have summarized important aspects of AI, so through which it is operating through machine learning, deep learning, natural language processing, and generative AIs. I think almost at present we are getting all generative AI models. So there are a lot of generative AI models in the market, which you can freely access also, and some are subscription based also. So let us see what is the impact of AI on pharmacy automations. So nowadays we heard about the intelligent pharmacy, so which will provide the automated care services. So we want to know how the artificial intelligence is transforming safety and efficacy across the modern pharmacy systems. So how they are affecting the patient's life care and other aspects. So artificial intelligence, it combines data. I think data may be from any sources, patient data or from hospital data or individual clinical data. Algorithms are certain programs which is, I think, written by various software professionals, combined with robotics to automate repetitive and high risk pharmacy operations. So, what are the advantages of this in pharmacy automation? This reduces manual errors in dispensing and verification. Nowadays, we see a lot of, I think, dispensing errors will be there. So, because of so many aspects there, so we can reduce the manual errors in dispensing and verification. So, if you make it fully automatic, it frees the pharmacist to focus more on the patient care. So, rather than focusing on the prescriptions and the contents in the prescription, so you can automate this operation and you can focus more on the patient care. So you can invest the time in patient counseling and giving the adverse reactions, possibilities, and what are the food drug interaction aspects, and this aspect you can make it. So which handles surging prescription volumes at scale, because sometimes in the larger pharmacies, I think number of prescriptions will be higher at particular time. So that also can be handled easily using the AI technologies. And also it will deliver the faster, safer, and more consistent services. So which is an advantage of this AI in pharmacy automations. So from counting trays to cognitive systems. So like what is the evolution of the AI in pharmacy automations? I think it started a long way back. So if you see in the 1970s to 80s, we got the first automated counting and dispensing machines, which existed already before around 40 years. But a slower progression happened in 1990 to 2000. So we got a barcode scanning. So that is also an automation type, and computerized verification, which already existed in 1990s to 2000s. So somewhere around 2010, so there was I think, a robust increase in all the machine learning technologies and all other related AI aspects. It started. So machine learning enters into clinical decision support. So that was, I think, benchmark of application of AI in your pharmacy automations. I think in 2020s, by the emergence of COVID-19, so robotics, computer vision, and predictive AI at scale, they have been developed. I think now a lot of research is going on and a lot of companies are coming out. So I think by 2026, at present, I think we have many things managed by artificial intelligence. And there are a lot of companies, they are investing into artificial intelligence technologies. So now there is a no limit, because if you learn AI properly and try to integrate it into a day by day work, so your work will be easy and you will be more efficient rather than doing everything manually. So there are some building blocks of automation. So what are these building blocks? So initially we need to have a robust hardware systems. So like we have dispensing robots, automated counters, packaging units, and barcode scanners that physically handle medications. And we need to have some software innovations also, where it will give pharmacy management systems, EHR integration, and cloud platforms are available nowadays that orchestrate workflow and data. Then very important is a lot of AI-based algorithms have been generated. So which serves as a model for pattern recognition, very important, prediction, and decision support that turn into intelligent actions. So it is through AI-based algorithms only the process is becoming nowadays very much easier. And you will be able to integrate this AI technology with any of your operating systems. Like you are using an Excel for some data making or some patient data. So you can automate the operations with the AI-based algorithms and make your work very easily, and productivity will be very higher. So it matters because 99% dispensing accuracy achieved through automated verification. So this has been proved. And there is a 3 times faster throughput, which is freeing staff for patient facing care. Okay, so that means the staffs can put their more time on the patient care rather than dispensing and other related activities. So lower operating cost from reduced waste and rework. So it will also reduce the unwanted waste and other thing, and also it will reduce the cost. That is very important for many of the things. So it will be having upper hand in cost reduction also. So overall, these methods gives the cost effectiveness in the operations. So how the AI is powering the pharmacies? So traditional pharmacies, if you see, so there is a pharmacist and there are one or two assistants will be there, and they will be involved in dispensing of medications. And most probably they could not handle if there is a more prescription surge at a time. So all these things will make the usage of AI techniques. So one is machine learning, so which learns from historical data to predict demand and flag anomalies. So like we have a historical data of patients. So if you have a patient data with their prescription and all the data is there, you can automate these prescriptions also, so that you can make your work easy. Natural language processing is reading prescriptions and clinical notes to extract structural data. Next one is computer vision, which verifies pill shape, if any deformities in the pill shape, if any variation in the color, and if any some misinformation on the labels or some informations are missing, so which will catch the dispensing errors. So that is also very important aspect of computer vision. Robotics can make the physical work less. So like physically picks, it will count and packages and medications with the precisions. So these are all a possible attribute to the pharmacies where AI can be used for automations. So next one is application like prescription filling and end-to-end operations. So NLP will read and digitalize the incoming prescriptions, and there is a computer vision confirms the right drug and the right dose, and dispensing and robotics count will be there. It will label and package it accurately, and pharmacists validate the flagged exceptions only. So you can only re-verify the flagged exceptions so that it will make your work easy. Then also it is a smarter stock and zero guesswork. So usually the guesswork is not allowed in this type of thing because it is directly related to the patient health. So tracking is easy. So real time, you can track real time visibility of every unit, batch and expiry date across locations. So this is one of the major challenges in segregating the medications based on their expiry dates. So whichever is near expiry, you have to segregate it and you have to make it more liquid. Optimization, AI balances stock levels to cut waste while preventing shortages. And also for forecasting, predictive models anticipate demand from the seasonal and prescribing trends. So based on the season, so it will help you to make the proper inventories. Suppose there is a season of rainy season is there, or some monsoon seasons are there, all infectious diseases and cough related, or there may be related to some aspects are required to be so inventory has to be maintained properly before the season starts. So otherwise there will be a problem with the stock holding and other aspects. So forecasting also can be done by the predictive models. So demand for the seasonal and prescribing trends by the various. So next one is safety and compliance. So that is also an important aspect of pharmacy automation. So automated drug interaction and allergy checks at point of dispense. So this is, I think, at this point of time, this point has a major role because nowadays there are a lot of drug interaction cases, which is seldom handled by the pharmacist because of his other work and is engaged in some other maintaining. So here you can also automate drug interactions also. So AI can easily automate what is a possible interaction between one and other drug, and also allergy checks at point of the dispense. So if the patient has a particular allergy or something, dosage validation against patient records to prevent overdoses. So that is also another parameter in patient safety. So sometimes there will be overdosing, which may be an error in prescription, or which are some other errors will be added there. So you can also go for a dosage validation against patient records to prevent overdoses. So all the AI modules will have already the patient records with one of the names or some identification numbers. So you can easily trace back the prescription and you can have a look onto it. And full audit trials supporting HIPAA and FDA compliance. So this can also be used to prepare the audit reports, like, and real-time alerts that flag risk before medication reaches the patients. So this is also one of the, I think, in the advanced healthcare systems in United States and European countries, I think this system has already in operation. So this is one of the major aspects so that it will definitely decrease the uh, error in medication and other toxic reactions to the medications. So what is the challenge and limitation? So I think India is a very big country and with the largest populations, second most population of around 140 to 145 crores. So implementing all this automated system may be a little challenge, but it can be operated through in phases so that slowly it can be implemented, starting from the metro, I think major cities, then going to the tier 2 cities, tier 3, then coming to the villages and other aspects. So it will take a very long time to fully automate our pharmacy systems. So one is technical problem will be there because legacy system integration, data quality, and high upfront cost. So cost is the one aspect because majority of the healthcare systems are not affordable with these modern techniques because a lot of investment is there at the initial time. And the data quality also very important because data quality matters very much because prescriptions and because many patients they don't undergo the regular uh, checkups. So once prescription is generated, they may not visit a physician for next to 3 to 4 years. So with the same prescription data, it's very difficult. So that's the one aspect. Ethical consideration also. So it affects the data privacy also because the patient's individual data. So there may be a problem with the data privacy also. Algorithmic bias will be there. Accountability problem for AI decisions. So that is also another aspect of limitations there. Then we have operational things like staff training is required. I think from the new syllabus, I think already that AI and machine learning, such type of subjects are already included. I think the future pharmacy students will definitely have a major uphand in these aspects. So majority of the challenge at present will be a staff training. So you have to train the staff for a particular AI applications, and workflow management and change is required. So there is a workflow modification is required, and dependence on system uptime is also required. And having a high robust internet systems, very high broadband systems of internet, so 24 hours it should be available. So such kind of things are little challenging and but can be overcome in the coming years. So we have some case studies where I think this modernization of AI in automated pharmacies have been an advantage of many operations like large hospital networks. I think even in India, we have a very large hospital networks. Maybe a few we can take as an example. So robotic dispensing, they cut medication errors by around 40% within 1 year. So this is the data which is generated from the by taking an example of the very large hospital networks throughout the world. So this 40% reduction in the error is one of the major, uh, I think, achievement. You can call it as an achievement. Retail pharmacy chains also nowadays we also see in India also there is operation of a large number of pharmacy retail chains. So central fill automation doubled the daily prescription capacity. So there is a centralized fill thing and their individual branches. So they will maintain an inventory in the central branch, and they can, at a given point of time, track the inventories of their subsidiaries wherever they are located. And it has doubled their capacity, prescription giving capacity. Then specialty compounding labs. So AI vision verification reached near perfect accuracy on complex doses also. So I think AI also it is going further modifications, and it is also becoming learning and machine learning also. It is continuous process. So data being, I think, feeded more, and there is also a probability of getting near 100% accuracy with AI models. So what is the next thing which has to be considered here in this case? So we have generative AI assistants for real-time clinical guidance. So that is one of the application of generative AI models for clinical guidance. We have fully autonomous micro fulfillment pharmacy hubs. So that is also a new thing which is coming up. And we have predictive personalized medicine and dosing. So personalized medicine is, I think, getting more importance in Western systems. I think slowly in our Indian system also it is getting adopted, maybe in bigger cities there. So I think in coming 5 to 10 years, personalized medicine and this type of things will be gearing up. So that is sure. So that will increase the quality of the patient safety and patient care. And there is a seamless interoperability across health data ecosystem. So you can talk with the various form of health operations. Like if you have a data systems which can integrate all the data into the system, so that will also have an advantage over health data ecosystems. So these are the things which is there with the AI in pharmacy automations. So how we can automate the pharmacy and what are the advantages and what are the future things we can expect from the pharmacy automations. Now I want to throw some light on the automation in pharma manufacturing also. So because pharma automation in manufacturing is gaining very much speed. So I think majority of the companies, they are adopting the AI systems for their regular, maybe their production control or maybe their quality control systems, because I think production involves, I think if it is, you take it as example of a tablet, so I think lakhs or millions of tablets are punched per day or something, depending on the capacity. So errors and other quality control systems at present they exist. I think they are not robust enough to, I think, check all the quality systems there. So I think many companies are adopting to the automation in pharma manufacturing. So this will involve the batch to continuous variations and schedule to productive, and AI-driven automation is reshaping the physical production of medicines. So this is Pharma 4.0, the smart manufacturing stack. So they have adopted some major actions here like IoT sensing layers. There are majorly 4 layers from sensor to decisions, and which define a modern AI-enabled plant. There is manufacturing plant. So we have IoT sensing layer, then data and historian layer, analytics and AI layer, and automation and control layer. So which is subdivided into 4 layers. So what is IoT sensing layer? So it is distributed sensors which stream temperatures, pressure, spectroscopic data, and vibration data continuously across unit operations. So sometimes in I think manual quality control, what happens is samples to be withdrawn from the particular batch as per the Indian pharmacopoeia requirement. So that has to be analyzed by the quality control department, and the report comes to the production team where. So it will have some time lag, but here continuous operation through temperature, pressure, everything is controlled. Spectroscopic evaluation is done and vibration data continuously across unit operation. So this will enhance the production capacity and also the productivity of a particular plant. So, and also it will reduce the chances of failure of particular batches. So initially, if it has been detected that there's some problem with the particular batch, it can be easily corrected in time. So that there may be any operational problems or anything which will not be affecting the production capacities. As you know, India is the hub for the, I think, generic drugs. So we are the, I think, uh, we are the highest supplier of the prescription medicines or generic drugs, particularly in the US market and other markets of the emerging markets in the world. So I think production capacity enhancement is one of the aspects of this AI. So through AI integration only, we can increase the production capacity by 2 times or 4 times of a particular uh, pharma entity, so that they can improve their production capacities and their performance. Analytics and AI layer will, so we have some machine learning models. They perform soft sensing, anomaly detection, and predictive quality scoring in near real time. So how much you train the model, so that much robust it will become. So we are training the model, feeding them particular data systems, particular same data systems. And before feeding that, the data itself has to be rechecked and revalidated. So such data can be used to train a ML model, so machine learning model, and predictive quality scoring in real time. So automation and control layer. So closed loop control and robotics, they act on model outputs, adjusting set points or flagging batches without manual intervention. So nowadays we have all automated systems which can sense, which can detect any minute errors in production layers, so that immediately the problem is identified and it is resolved at a particular point. So otherwise, I think human intervention will not be sufficient in this case if it is a very large production operation, and so AI can easily help in that. So let us go for application of this automation or AI technology in analytical technology also, PAT, and real-time release studies. So we have like inline spectroscopic studies which can be integrated with AI models. So we have near infrared spectroscopy, Raman, which probes, monitor blend uniformity and reaction progress continuously rather than via destructive sampling. So you cannot use a destructive sampling in this case. Continuous in operation, we can easily take out the sample and we can easily analyze the sample. Then we have chemometric tools plus machine learning models, so multivariate models which translate spectral signals into concentration or quality estimates in real time. So I think this is a very good application of chemometric tools plus machine learning models, so that real-time spectra you can get, and you will get the impurity profile or whatever the information is there. You can take it, and so this will help you in reducing the human intervention. So real-time release testing, like sufficiently validated PAT models, allow release decisions without a full end of line lab assay, cutting cycle time. So continuous manufacturing fit. So PAT is a prerequisite for continuous process. So wherever there is a continuous process happening, the PAT is a very essential requirement where there is no discrete batch to sample after the fact. So regulatory anchor is, so PAT and RTRT are formally recognized in ICH Q8, Q9, Q10, Q13. So they are aspect of the quality by design guidance and FDA's PAT framework. So I think now US FDA approval also based on integration of all these newer AI methods integrated into the manufacturing service and get the proper data, which is also generated during the process. So AI-based soft sensors used to release decisions. They must meet the same validation rigor as any GMP critical instrument. So nowadays already GMP certified labs and GMP uh, certified manufacturing units are there. Also, they are going for FDA approval. So integration of the AI-based systems will definitely help them for getting the FDA approval, and definitely inspection formalities and all. They can easily clear it with implementing all these AI techniques. So predictive maintenance, like reactive to foresight. So reactive means it is fixed after failure. That means highest downtime and unplanned batch loss. So that is a reactive. Preventive is fix on fixed schedules, like reduces failures, waste, remaining component life. So predictive AI will fix when data indicates need, which minimizes both downtime and unnecessary servicing. So this is the integration of the predictive AI in both the reactive as well as preventive measures. So framework adopted from general reliability, engineer literature, validate against our own sites and data before quoting specific saving speakers. So now robotic process, I think robotics are one of the major stakeholders now because of the automation. So many of the pharma industry they go for the robotic operations wherever is required. So wherever critical human intervention is not required, so all the operations are run through the robotics. So automation extends past physical robotics into the paperwork and data flows that surround production. So we have physical robotics. Robotic arms handle aseptic filling, so which is major required as per the Schedule C, and these are required packaging, material transfer with the sub-millimeter repeatability. So batch record automation like RPA boards reconcile electronic batch records against SOPs, flagging deviations for human review instead of manual line by line checks. So these are the advantages. Deviation and kappa trade. So NLP models cluster free text deviation, which reports surface recurring root causes across sites. So you can integrate into across sites where the manufacturing process has been taking place and integrate them into a one. So this is a major advantage of this. So demand linked scheduling like AI driven planning tools. So production runs against forecast, demand, and changeover cost simultaneously. So demand linked scheduling. So I think majority of the time this is a major challenge for a pharmaceutical company because you don't know how much production is needed and how much demand is there in the market. So based on the seasonal things like and the requirement of particular. So in some region, infectious diseases will be highest in particular months. In some regions, that digestive and related problems will be there in particular time. So in that case, I think production enhancement will also be a challenging. So all this can be automated, and AI-driven planning tools will control the production and forecast the demand and change our cost simultaneously. So this is a major advantage of this forecasting. So AI in supply chain and demand forecasting, like why this majorly important for the public health. So this is majorly related to public health. So accurate demand forecasting is not just a cost issue. So stockouts of essential medicines and vaccines are a direct public health risk. So any point of time, there should not be a stock out of medications, essential medications, and in particular time vaccines if there is a emergency is there. So AI-based forecasting blends historical dispensing data, epidemiological signals, and even social media or search trend indicators to anticipate demand shifts like seasonal antibiotic demand, outbreak driven vaccine surges like COVID-19, then earlier than classical time series methods alone. So this classical time series method may not be applicable. So in India, actually Indian scenario, this may be more significant because the consumer, this one is very high. So consumer demand is very high in India. So this will help you in maintaining the forecasting of a particular, and you can also, there is a demand in production also, you can increase the demand or decrease whenever the chart says whether to increase or decrease the production. So that is a major application in forecasting. So how much to be produced and how much is required by the market and how you can maintain these inventories. So next is very important is computer vision for quality control system. So sub-visible defect detection at production line speed. So sometimes there are very minute defects which cannot be found out through the production line. So whenever it reaches the market, I think through some patient who is taking the prescription, so he may notice a minor changes in the thing, like there may be some discoloration in the tablet, there may be damaging of some strips, there may be particles lying in the injections or injectables, and sometimes physician will identify some abnormalities. So convolutional and transformer based vision models inspect tablets, vials, and blister packs for cosmetic and structural defects, chips discoloration, fill level anomalies, and foreign particulates. So there are important aspects of quality control. So at speeds and consistency level, manual visual inspection cannot sustain across a full shift. It cannot sustain. So what are the advantages? Which removes the inspector variability, and fatigue-driven misrates in manual visual quality controls. So this requires a large, well-labeled defect image libraries, including rare defect classes, to avoid blind spots. So what we can do is, already there are some defective tablets or blister packs or whatever is there. All these images can be captured and it can be given to the model to learn. So the model will learn itself and it can easily identify. Otherwise, blind spots and other thing, it can also give error. Even though the packing is perfect, sometimes it can give error also. So that is the thing you have to take care. So model performance must be validated against confirmed defect ground truth before replacing the manual sign off. So before replacing any manual operations, we should time test the AI generated process. It has to be repeated. So it has to give all the values similar in any circumstances. So you should have proper this one validation also of this matter. So validation is very important aspect in this control. So next section will be that application of artificial intelligence in clinical trials. So I think this is somewhat related to the public health analytics. So this also helps in clinical trials to make a decision on how to recruitment, how to make an adaptive design, how to make a safety monitoring, applying AI where cost and time pressure are most acute. So wherever you have a time pressure and cost pressure, so slowly the AI models can be integrated into the process of clinical trials so that it can be made effortless. So the clinical trial bottleneck is trial remain the single most expensive and time intensive phase of the development and the phase most exposed to recruitment failure also. So sometimes there is a recruitment failures and a lot of possibilities are there. So typical trial duration across the phases extended further by slow recruitment. So sometimes clinical trials are not finished by a particular time barrier. Sometimes they get extended also because of the recruitment failure, sometimes withdrawal of the patients and other thing. Trials that miss enrollment timelines. So industry-wide estimate verify against current CRO benchmarking report. So the report says that sometimes there is a miss enrollment timelines in clinical trials, and cost of each month of delay is very high because the clinical trials are very expensive and it has to be managed within that particular time and within that particular budget. Compounds across staff, site, and opportunity cost. So these are all adding into the clinical trial bottlenecks. So what is the application of the artificial intelligence in patient recruitment and site selection? So matching the right patient to the right trial is fundamentally an information retrieval problem at scale. So sometimes we don't know that which type of patient has to be recruited for which type of trial. So that itself is a fundamental problem. So for that we have EHR based cohort matching like NLP over unstructured clinical notes identify patients meeting complex inclusion exclusion criteria, which is far faster than manual chart reviews. So this is the advantages. Site feasibility scoring. So machine learning models rank candidate sites by historical enrollment performance. So you can take a data from the historical enrollment of a particular clinical trial and take the data and the catchment demographies you can run and protocol complexity fit. So if you have a particular data and you have already trained the machine learning model, so it can easily identify which type of patients will be suitable for which type of clinical trial. So diverse cohort optimization. So predictive models, they flag under recruited subgroups early, supporting representativeness rather than the convenience sample bias. So next one is patient facing matching tools. So conversational agents help patient self screening against plain language eligibility criteria before formal. So these are all advantages of the recruitment of AI in clinical trials for the following applications. So adaptive trial design and synthetic control arms like classical fixed design, which goes on sample size and arms fixed at protocol design. Interim looks limited and pre-specified sparingly. Every arm requires a concurrently enrolled control, and underpowered subgroup signals often go undetected. So what is the advantage of AI-enabled adaptive design? So we have Bayesian classification models and machine learning models. They will re-estimate sample size mid-trial. Frequent interim analysis with pre-planned stopping rules, synthetic control arms built from historical or real-world data, and continuous subgroup response modeling flags responders early. So these are all the advantages once you adopt a AI-enabled adaptive designs. So real-world data and real-world evidence like data sources. We have databases like disease registries and variable derived data and raw material for RWE. Casual ML methods like propensity score matching, target trial emulation, doubly robust ML estimators. Post-market surveillance also we have. RWE extends safety and effectiveness, monitoring well beyond the controlled trial population and duration. So also you can automate this for post-market surveillance, which is very important phase of the drug. So that is the phase where majority of the drawbacks of the given drug will be definitely uncovered. Regulatory acceptance. Agencies increasingly accept RWE as supportive evidence for label expansions and post-approval commitment. So RWE is only as trustworthy as the confounding control strategy behind it. So this topic should be like case study. Whoever is indulged in teaching of this clinical study and automation of clinical trials, so you have to take care of this bias. So that has to be taken care. So even AI also can be integrated in pharmacovigilance. So as you know, pharmacovigilance field is, I think, it's expanding, and a lot of newer companies are entering into it for pharmacovigilance. And also, it is one of the placement opportunity for many postgraduates, even in the undergraduates also. So pharmacovigilance, like post-approval safety monitoring, is increasingly an NLP and anomaly detection problem operating on massive noisy text stream. So automated case intake like NLP extracts structured adverse event data. So I think nowadays everybody is maintaining the data on adverse reaction, and at least in the major hospitals where pharmacy practice wing and other things are there, they are maintaining it. So data can be taken from the any sources. Signal detection. So disproportionality and bias in data mining algorithms, they flag drug event pairs occurring more often than chance across the spontaneous report databases. So social and digital listening, so monitored consented data streams supplement formal reporting channels, surfacing signal earlier in some cases. So case triage and prioritization. So ML scores route the most clinically significant or serious cases human reviewers first. Okay. So next is AI in regulatory and quality systems. So I think now regulatory affairs is one of the field where so majority of the um, system will see the regulations. So whether it is a generic drug regulations or new drug regulations. So regulatory affairs become the important aspect. Now, a lot of AI integration is happening in regulatory affairs. So AI moves from the research tool to decision support in GXP settings. So regulatory frameworks and validation practice must keep pace. So a lot of data will be there in regulatory system. So application of AI will definitely help in maintaining this system. So regulatory landscape for AI and artificial intelligence and machine learning. So software as a medical device like FDA's particular AML based SAMD action plan and predetermined change control like emerging guidance, specified model updates. Good machine learning practice also been established, GMLP. Cross agency principles cover data quality, model transparency, and performance monitoring. Okay, covering established GXP culture. Nowadays, there is also a check for good machine learning practices also, because everything is data driven. So all the outputs you are getting based on the quality of the particular data. So that's why it is very important. So regional variation like EMA, FDA, and other regional bodies are converging on similar principles, but differ in submission specifics like always confirm current local guide. So even the FDA, MHRA, or any other agencies, they are also recommending to implement certain AI models or machine learning based models so that your process will become easier. So this is a fast moving regulatory area when teaching direct students to check FDA and EMA websites directly for the current version of any AML guidance document rather than relying on a static slide. So these guidelines and other things are very much changing, I think, time to time. So rather than referring to any static guidelines, so the dynamic FDA and EMA websites can be encroached and you can get the realistic uh, data, what is happening actually in operation, and you can easily update it with the latest happening. So explainability and model validation, like interpretability of methods. Okay, we have visualization help domain experts audit why a model reached a given prediction. So that is also possible. Cross validation, performance monitoring, ongoing drift detection, catches silent degradation as input data, shift post deployment. So documentation traceability. So model cards and data lineage records support audit trials comparable to existing GMP documentation practice. Nowadays, our GMP documentation practice also recommend usage of the AI models, but only one condition is all these things has to be cross-validated. It should be a robust model predicting them. So human in the loop sign off. So high stakes decision retain a qualified human reviewer. Ultimately, whatever the AI generated models or whatever the AI generated details or documents are there, the final decision only will be taken by a qualified human reviewer as a final accountable party, not the model alone. So this is the advantage there where the job, okay, so human jobs are not replaced here. So ultimately, whatever models are generated, everything has to be checked by a experienced person, and accountability and model is not alone will be responsible for deciding on the integrity or accuracy of a particular data. So if your data input is proper and machine learning models are accurate, then definitely you can get a very high success in predictions. So regulatory automation, there are some cases where dossier drafting and compilation. So they are using large language models or NLPs. Primary benefit is faster first draft assembly for CTD modules. Literature surveillance, so NLP plus information retrieval, so continuous monitoring for label relevant new evidence. Submission quality control, rule based plus ML consistency checks, catches cross reference and formatting errors pre submissions. Labeling comparison can be done, text similarity models, flags unintended deviation across regional label versions. Inspection readiness, anomaly detection on quality records, and surface documentation gaps before a regulatory inspection. So these are all the applications where the AI models being trained and used for particular benefits. So the major thing of this topic will be AI in public health analytics. So there is a part of the title there. So shifting the lens from the individual patient to the population. So nowadays we have to consider a total populations as not as an individual patient. So surveillance, forecasting, equity, and resource allocation. So they are all can be done with the utilization of the AI. So the public health data ecosystem. So population level AI draws on a wider and a messier set of data streams than the clinical AI alone. So clinical and administrative data like hospitalization records, notifiable disease reports, and insurance claims anchor traditional surveillance. So genomic surveillance like pathogen sequencing, data tracks variant, emergence, and transmission chains in near real time. Environmental and mobility data, climate and water quality and other aspects. Digital and participatory data like search trends, wastewater signals, and app-based syndrome reporting. add early if noisy signal is there. So AI in disease surveillance and outbreak detection. I think after the emergence of the COVID-19 pandemic, so even there is a usage of the AI-based models for outbreak detection, and in that particular emergency cases, this can be deployed, and so we can make a use of that core applications like detecting the signal before it becomes a headline. So anomaly detection models continuously compare incoming case counts, syndrome indicators, genomic data against expected baselines, flagging statistically significant deviation for the epidemiologist review well before a cluster would otherwise be manually noticed. So these AI models can give an initial or earlier, I think earlier warning or signals to the epidemiologist, so he can review it before a cluster would otherwise be manually noticed. So time series anomaly detection, spatial scan, ML-based ensemble forecasting. So strength is like scales across thousands of reporting units simultaneously. So that is the advantage of that. So caveat is signal quality is bounded by the underlying reporting completeness and timeline. AI cannot fix upstream data gaps. So data integrity and data creation and making a particular well versed or highly accurate data is responsible for is a responsibility of the individual. So whoever group reporting the data particular, so if they report a particular data in a systematic manner, it is easy for the model to grab it and to make use of it for variety of purposes. So predictive modeling for epidemics like mechanistic models are available, pure machine learning forecasting, hybrid mechanistics using machine learning and ensemble forecasting. So pure machine learning models can overfit to a specific outbreak shape and generalize poorly to a new pathogen. So pure mechanistic model can be too rigid to capture behavioral or reporting shift. So hybrid approaches are increasingly the practical middle ground taught in modern epidemiology curriculum. So illustrative epidemic curve like model versus the reported cases. So here, so one line says it is a reported cases and one is predicted by the model. So this is a synthetic illustrative curve, not the real outbreak. So it is walked through the forecast evaluation concept. I think this is one of the model which is being generated between the two. So the peak and the turn down even when overall trend capture is strong. So there is a systematic way where the model is predicting a particular this one. So you can see there is a almost a matching between these two lines. So there is an important case study. I think during COVID-19 response, so AI based models were extensively used. So stress test for population like scale AI at unprecedented speed. So the pandemic compressed years of AI in public health adoption into the months. So I think AI was already being developed from the early 2000, 2005, but it was not adopted into the public utility. So, but pandemic has brought the health adoption into months. So within months, it was being adopted. The genomic surveillance pipelines tracked variant speed, mobility informed models, supported non-pharmaceutical intervention planning, and machine learning assisted triage tools supported overloaded hospital systems while also exposing real limitations. So because of that, you could know some aspects of the limitations. So rapid genomic sequencing pipelines and dashboard-based situational awareness at scale. So several widely publicized clinical prediction models later showed poor external validation and hidden bias. Speed of deployment and rigor of validation are in constant tension, a core discussion point for training. So this is the thing. So next is AI in health equity and social determinants. So population models are only as fair as the data and objectives they are built on. So whichever data you are training the machine model, ML, so particularly when the data is proper, so then only you can have a robust model. So representative gaps are like underrepresented populations in training data lead to systematically less accurate predictions for those similar groups. Social determinants as features like housing, income, and other aspects. Fairness-aware modeling techniques such as subgroup calibration, constraint can build in the model objectives. Community-engaged design like involving affected communities in problem framing improves both model relevance and public trust. So that is also important. So geospatial AI and GIS public health, like ML models combine environmental, demographic, and case density layers. Vector-borne disease modeling, satellite-derived climate, and land use data feed models. Resource access analysis, geospatial models identify healthcare access. So outbreak source tracing, like spatio-temporal clustering algorithms, so they can detect during certain investigation. So there are many open source tracks are there. So nowadays there are many are there, so it can be utilized by the healthcare professionals. So health policy and the resource allocation also your AI being used, because scenario simulation, agent-based and compartmental simulations let policymakers compare intervention strategies before committing resources. So workforce and bed capacity forecasting, vaccine allocation optimization. So I think majority of the COVID-19 vaccines, so properly it was being distributed in each corner of the country. So maybe utilization of particular models. So optimization model balance, equity and efficiency objectives when distributing limited vaccine supplies. Budget impact modeling. So ML informed health economic model support cost effectiveness analysis for programming funding decisions. Also, it can make a decision on funding and other related aspects. So wearables and IoT and digital phenotyping. So continuous physiological streams, heart rate, activity, and sleep data from consumer wearables provide passive population scale health signal, aggregate syndrome signals, then privacy by design requirement, and chronic disease monitoring also can be done, like continuous glucose and cardiac monitoring data, which support both individual care and population level chronic disease surveillance. So these are the application in public health aspects. So cross-cutting themes like governance, ethics, and workforce readiness are not addenda to the technical story. They determine whether any of it can be responsibly deployed. So this is very important in implementation of the artificial intelligence for this one. So one of the things are data privacy, security, and governance. So health data sensitivity and re-identification risks make governance a first-order design constraint, not a compliance afterthought. So regulatory frameworks are very important, like HIPAA, GDPR, India's DPDP Act, and comparable regional laws each define consent, minimization, and breach notification. So de-identification limits, then federated and privacy-preserving learning, and data governance structures. So institutional data access committees and audit trials remain essential even where the technical safeguards are strong. So time to time, there is a revival of the data, and all the AI-based models has to be monitored regularly. So there should not be any other aspects of this, because a lot of data is being captured. So workforce and skills preparing faculty and students. So computational literacy. So basic fluency in data handling, statistics. I think this is a prerequisite for the pharmacy graduates at present time. So already being facilitated in the new syllabus, where already they're being introduced to AI, Python-based languages and programming and machine learning techniques. So they are learning a lot about the AI technologies in their graduation. So domain AI transition skills, like highest value skill is often translating domain problems into well posed. So students can be given smaller projects on the AI and other applications, so that they can be get themselves readiness for the AI application. So interdisciplinary collaboration. So effective teams pair domain experts with data scientists. So neither substitutes for the other. So I think data science is also emerging along with the pharmacy people are, I think, making use of the more AI and other things. So it can be integrated. It is a multidisciplinary area, because even clinicians pharmacists and data scientists can come together, so help in building models which can be accurate and robust. So domain AI translation skills are their continuous updating. So given the field space, a habit of ongoing primary literature engagement matters more than any single fixed curriculum. So even students should be well versed in the current literature. So whatever the progress is happening in AI, in particularly healthcare and medicine, so that they can be updated with the latest applications of AI in healthcare. So that is very important aspects. So curriculum integration ideas, like so those who want to implement this thing in their regular curriculum. So I have given a list, I think, where type of models can be used in which type of subjects or their domains, right? I think medicinal chemistry, they are already using these AI models, drug design, QSAR, uh, everything. So critique on AI proposed scaffold for synthesizability, and pharmacology people are learning ADMET, absorption, distribution, metabolism, excretion through ML models. So compare ML predicted versus experimental ADMET data and make a data file. So pharmaceutical technology people, PAT and continuous manufacturing walk through a real-time release decision tree. So which is artificial neural networks and other related aspects. Clinical research method involve adaptive trial design like simulate and interim analysis stopping decisions. Public health and epidemiology, the people who are working on public health and other aspects. Outbreak forecasting exercise like build a simple SIR model and compare to an ML baseline. So research ethics, bias and fairness case study, debate the cost as proxy algorithm case in small groups. So these are all adaptive techniques you can integrate into the curriculum. So along with the regular to the syllabus which is there. So that will enable the students to adopt to new AI techniques, which is I think compulsory requirement nowadays. So without any AI domain, it is not possible to work in any one of the aspects. So that's the thing. So, but there are some challenges and barrier to this adoption. So data quality fragmentation. So inconsistent data, incomplete data. So this is another problem there. Infrastructure cost. Computing is itself is very costly because GPU enabled systems are required for machine learning approaches. Licensing using the softwares, integration cost or other public health sector agencies. Regulatory uncertainty, evolving guidance creates hesitancy to deploy AI in decision critical workflow. And trust and change management. So clinician and researcher trust must be earned through transparency and demonstrated reliability, not assumed from the technical, the field which we are closely working with. So then only it can be implemented. So what are the outlooks? So where the field is heading, what to carry back? So teaching and research and questions. So futures outlook is like foundation models for biology. So this I think computational biology and other people. Agentic AI workflow for multi-step autonomous agents for APIs and moving phenotype to pilot deployment. Federated privacy preserving analytics like cross-institutional model training without centralized data sharing address both privacy and data fragmentations. Real world post market data increasingly feeds back into discovery and manufacturing decisions, closing a loop that once ran one way. So the ultimate message is AI does not replace any of these chemist, the trial statistician or an epidemiologist, but it changes what each of them spends their expertise on. So the recurring theme across every section today is like AI compresses search spaces and surfaces patterns at a scale humans cannot match. So this is the only thing where the AI is getting advantage because it can process a huge data in no time. So, but what is the quality of the data which feeded? So AI cannot make own decision. So whatever we feed, it will give its data based on the original data. So this should be taken utmost care, the quality of the data systems. Like while judgment, validation, and accountability remain irreducibly human tasks. So humans cannot be replaced in these decision making steps. So thereby human intervention is definitely required. So AI is not superior to humans. So AI is only an agent which we can use, and we can make the work very simple, and productivity can be enhanced. Okay. So for references, I think we can take under each segment like manufacturing, regulatory guidance, literatures, and ICH Q8 to this Journal of Pharma Innovations, clinical trial AI, so clinical search methodology journals. These are the journals. Public health analytics, epidemiology, and public health informatics. I think a lot of articles you will get nowadays for AI integration in healthcare. So there are n number of articles are available. So you can easily read it and you can get understand by all these aspects. Interdisciplinary ethics and policy, like all these journals will definitely have this type of articles. You can download an individual article or a group of articles and you can update the recent progress happening in AI in healthcare, that is pharmacy automations and public health analytics. I like to thank you very much for providing me an opportunities for giving a talk in this faculty development programs. So innovating pharmacy education with Python, AI, machine learning, and psychology. So which is organized by School of Pharmaceutical Education and Research, Jamia Hamdard, New Delhi, in collaboration with the P.R.S. Education Trust and Department of Pharmacy, so LLRM Medical College, Meerut. So I like to thank all the organizers, organizing committee members, so all the faculty and heads of the pharmacy institutions, and all the faculty members and all the participants who participated in this faculty development program. So thank you very much, and as a special thanks to Vibhu Sahani sir. So thank you very much. For any questions are there, you can ask.
Thank you, Dr. Arvind. Thank you very much. Thank you very
Yes,
much
sir.
for sparing time for us. And
Thank you, sir.
really, it's very
My pleasure.
informative. Yeah, very informative session. And thanks a lot for joining.
Yes, sir.
Wishing you all the very best and convey my all the regards to for the Corps Manipal.
Yes, sir. Yes, sir. Yes, sir. Yes, sir.
Yeah.
Definitely, sir.
Thank
So
you
any
so
questions,
much,
sir?
sir.
My photo, my actually my photo is not coming. I don't know why some technical issue is there. Even though I have not stopped the video.
Okay, sir.
Yeah, now it has come. Yeah, sorry.
Yes, sir. Yes, sir. Yes, sir. I can
Yeah.
see.
Thanks a lot. Thanks a lot.
So thank you, sir. Thank you very much.
Convey my regards to Mutalek ji also.
Yes, sir. Yes, sir. Sure, sir. Sure, sir. Definitely, I'll convey.
Yeah, good.
Yes, sir. Have a nice day, sir. Any questions are there from participant?
Yeah, any question from the participants?
Actually,
We
sir,
Yeah,
we do not
tell me.
have any question regarding the session.
Okay,
So we
okay.
will wind up here. Thank you very
Yeah.
much, sir, for such an insightful and inspiring session. Your enlightening presentation on AI in pharmacy automation and public health analytics has provided us with valuable perspective on the transformative role of artificial intelligence in modern pharmaceutical practice and healthcare system. We sincerely appreciate you for accepting our invitation and taking the time to join us today. Your expertise, valuable guidance, and thought provoking deliberation have greatly enriched this faculty development program. As a token of our gratitude and appreciation, we are pleased to present your certificate of appreciation and a virtual memento on the screen.
Thank you. Thank you very
So,
much.
this is your
Thank
certificate,
you very
sir,
much. Dhanyavad.
and this is the memento, sir. The soft
Thank you.
copy of this token of appreciation will be shared with you soon on your email, sir. Once again, thank you so much, sir, for your presence, your valuable time, and outstanding session. Thank you so
Yeah,
much.
thank you. So again, thanks to all the organizers and Dr. Akhil Sharma sir and Dr. Vibhu Sahani sir. So it is I'm very excited to see all of you. So
Thank you very much, sir.
pleasure for me. Thank you, sir. Have a nice day.
Same
Have a
to you,
nice
sir.
day. Thank you.
Everyone, we are concluding the day 4 of our faculty development program. We reflect on another day filled with enriching discussion and inspiring learning experience. On behalf of the organizing committee, I extend our heartfelt gratitude to our esteemed speakers, Professor Dr. Zinat Iqbal and Dr. Arvind Pai sir, for sharing their exceptional knowledge, expertise, and valuable experience. We sincerely appreciate your precious presence, your time, and your inspiring contribution to this faculty development program. Tomorrow marks the final day of our faculty development program. We look forward to your enthusiastic participation as we conclude this wonderful journey of learning and innovation. Until then, thank you once again for being with us. Have a good day, and we look forward to welcoming all the participants tomorrow at 11 a.m. Thank you, everyone. See you tomorrow for the final day of FDP.